Hello and welcome to our Boston Biolife meeting. My name is Joseph Krieger, and I want to take a moment to thank you for coming in attendance at this exciting meeting. Boston Biolife 2021, we have exciting programs that we'd like to let you know about. So in the 2020 season, we were able to pivot and create a lot of educational content around our webinars, our industry-supported webinars. So we ran over 324 webinars and over 60 different topics, and we supported over 10 other industry events. We are introducing the concept of the Boston BioLife mini courses, which you're listening to now, and we've launched our MedPub research tool, as well as adding enhancements to the Boston BioLife Academy. So in 2021, we'll also be offering new programs such as additional cutting edge webinars, enhanced mini courses such as the one you're listening to today. We have physician incentive programs, the preferred premium provider platform. I know that's a lot of P's, but really what it is, it's a lot of value for healthcare providers that rely on Boston BioLife for educational content, either in the form of educational components like this or our research profile, using the MedPub research program and having access to critical industry events and other offerings. The healthcare provider programs for Boston BioLife are coming up include, again, the webinars that you're listening to or used to listening to. We should have at least 2 of these a week now, and they fall within the context of biotechnology or the biochemistry, clinical applications in clinical practice, as well as implementation of these in using practice management techniques. Our mini courses, such as the one you're listening to now, are becoming ever popular, and what we're doing is combining 3 or 4 speakers on a very concise topic so that we're not going into the long weekends or spending hours in front of the computer. Remote learning has a lot of benefits, but one of the time constraints. So with this is also consumption at your own pace, as these can be are looked at later as well. Some of the topics that we'll be covering are important, especially in the recent COVID-19 initiatives such as immune health, looking at the role of gut health, neurological wellness, personalized testing, medical imaging, and functional medicine, just to name a handful. These will be growing as we go forward over the year. The preferred premium provider program will offer healthcare providers the ability to have access to our cutting edge topics from our academy, such as our 11-part series, Introduction to Regenerative Medicine. This is a great way for people to get a good orientation to some of the terminology, nomenclature, derivatives orientation, and clinical applications of these exciting technologies. And you also have access to either our aesthetics or our pain management program, and access to 4 mini courses coming up as we develop them in the 21 season. This is access to peer-reviewed journal articles from major journals around the world covering different topics that are very relevant to clinical practice. The MedPub research tool comes with 20 CME credits, covers over 9,000 peer-reviewed journal articles, and again, it's an easy way to stay current. You can copy, print, and share the papers and articles. You can become involved in best practices. They function as a desk reference and also can be used for patient education and patient engagement. Boston BioLife has been working with several partners in industry to provide you access to other meetings such as the AAOM, the American College of Regenerative Medicine, International Cellular Medicine Society, Total Orthopedic Institute, Orthobiologic Institute, and the World Academy of Pain Management Ultrasonography. The World Stem Cell Summit will be having a meeting this June in conjunction with Wake Forest, and that's always a good place to see the cutting edge research and science. So discounts are available to our members and our listeners to attend these exciting meetings that give you a different perspective on the world of regenerative medicine, translational medicine. And as always, I think it's a good idea to look at other organizations to gain proper perspective. As I said, industry access gives you discounts on other industry events, and you can stay compliant with resources. And you also have the ability to have incentives from our technology providers and some of the training that they offer as well, which is always a good thing to have available to you in your practice. Our industry access also provides you information from a wide variety of technology providers. This is a small list of the people that have been working with Boston BioLife, and they cover many of the different products that are available on the spectrum of medicine and regenerative medicine. So we appreciate you supporting our vendors. Without them, it's difficult for us to have these programs and to stay current with the latest innovations from the market. So today's program is the spotlight on regenerative medicine for hair restoration. We have a very exciting program with industry leaders that have really been involved in pioneering these sciences as well as applying them in clinical practice. And we'll be looking at many facets of hair restoration, hair loss, and the biochemistry of the science of hair during today's presentation. So here's our schedule and our agenda, the topics. So my name is Joseph Krieger. I'll be moderating. I'm the president of the Boston Biolife. The first presentation will be the Biochemistry of Exosomes, a really new perspective by Duncan Ross, PhD, and the founder of Kimera Labs. Duncan has a large body of evidence and experience in not only immunology, but in the biochemistry of cell signaling and exosomes. As you know, the pathophysiology and trichology of scalp health will be conducted by Misti Barnes. She's a practicing trichologist and she does personal training and has a wide variety of experience in peptides, plant-derived biologics for hair restoration. Dr. Yael Halas has been practicing hair restoration technology in conjunction with plastic surgery in the New York area, and she's recently published a paper which we'll be making available for you as well. I think you can see it as part of the link to this meeting. And then finally, Dr. Alan Bauman, who's really a pioneer in the concept of hair restoration, and he has the Bauman Hair Hospital and has been really leading the way in the approach to hair restoration and looking at all the different facets that come together to help you not only maintain the hair you have, but also restore it in the event that you're losing your hair. So Dr. Bauman, we're very pleased to have him with us today. And at the end of the program, we'll be having a live question and answer with the panel. Many questions have come in already, so we'll do the best we can to address the questions that you have, and we appreciate the input from the audience in helping us build this program today, and thank you so much for your time and attention. So with that, we'll begin our program. So the first presentation will be from Mike Carroll. He's the VP of Business Development, and he'll give us some insight into Kimera Labs. So Mike, thank you very much.
Hello, everyone. My name is Mike Carroll. I am Vice President of Business Development for Kimera Labs, and I want to formally thank you for joining us today. I want to share some information regarding Kimera, the company, and our products for those of you who may not be familiar with us. First of all, Kimera is a producer of exosome products. We are not a distributor. All products are produced in our 27,000 square foot lab in Miramar, Florida. And that distinguishes us from some of the other companies in the exosome space who actually do not produce their own products. In addition to that, to the best of our knowledge, we are the only exosome producer whose lab has been fully audited and inspected by the FDA with a no action indicated response. Regarding our products are unique in that we produce our exosomes from a master cell line that was derived from a single donor placental tissue source. From that master mesenchymal stem cell line, we produce working cell lines that we use to produce our exosome products. So we can confidently state that our exosomes are 100% mesenchymal stem cell derived because they're produced from a master cell line. And this is important for a couple of reasons. First of all, from a potency standpoint, but secondly, as all companies in this space seek regulatory approval, we are going to have to demonstrate to the FDA that we can produce a pharmaceutical grade product in a reproducible manner. And we believe the only way to do that is to produce our products from a master cell line. We use the youngest ethically available source to produce our products, and again, that is a single donor placental tissue source. We utilize a proprietary isolation and purification protocol, and more than 40,000 patients have been treated with Kimera exosomes in the course of the last 5 years, and we have never had a reported serious adverse event. In addition to that, we have filed our first IND application with the FDA, to treat moderate to severe ARDS secondary to COVID-19. And we expect to hear back from them very soon as to when we can start our first clinical trials. That will be the first of several IND applications we plan to submit prior to the FDA's new May of 2021 deadline. Again, we thank you for your time on the webinar today, and I'll turn it back over to Joe to get things started.
Thank you, Mike. As always, we appreciate hearing from Kimera Labs. You know, you guys have been out there, developing these technologies, and you're really the pioneers in the field. And seeing the transparency that you have and the efforts that you're making to let people know how your products are created and the regulatory steps you're taking. And I know Duncan's worked tirelessly to create other quality control measures, and he'll be describing that as well during his presentation. So the first presentation, I have the honor of introducing Duncan Ross, PhD. He's been the scientist, in my opinion, that has contributed the greatest body of evidence and research in his own personal experience, and now with the Kimera Labs, that he's been working diligently to create the next generation of therapeutics in using exosome technology and really looking at the pathophysiology of these diseases. And as the founder of Kimera Labs and its chief scientific officer, Duncan, thank you so much for once again being part of Boston Biolife. We always look forward to your inspirational talks and your leadership in the field.
Hello, everyone. My name is Duncan Ross, and I am the CEO and founder of Kimera Labs. Welcome back to another Boston Biolife event. I've been with you here for many years, and I'm very excited to present to you what I have following. It's a new topic that I haven't really discussed before. It's hair loss. I don't want to be contrite, but it's not something I've spent that much time thinking about. But in researching this particular presentation, I've really become very excited at the possibilities, and I'm more excited at the fact that really resonates with my interest in autoimmunity, inflammation, and regenerative medicine. They all come together in this specific topic. So if you'd like a hands-on demonstration of what we're discussing here, because really we're going to get deep, but we can go much deeper. So if you'd like to join us on March 26th in Los Angeles at our offices there, or on April 17th at our laboratory in Miramar, Miami, we can go over these things in further detail. So I had COVID a few months ago. It wasn't extremely scary for me, but it was very scary for my mother. We both had it at the same time. I had to institutionalize her. I put her in the hospital for 5 days. They took wonderful care of her. I tried to take care of her for 3 days myself, and it was a dismal failure. I'm not a nurse, and I don't have a lot of patience. But the different methods that we had, and I was receiving from all of you, were varied. But when she got to the hospital, she did just fine within 3 hours. She didn't have any sort of pulmonary involvement, though. But what we're finding, though, and we've been receiving, is that once people are leave the hospital, either they were in the ICU and they leave with a PaO2 level of about 92% oxygen, and really you don't feel good at that point. You want to be 96 and above. So we've seen these types of patients. You may or may not know that we filed an IND for treatment of patients after COVID. One hiccup we ran into is that we wanted to treat people multiply on days 0, 2, and 4. That's 3 infusions. Because the inflammation from COVID is so strong. The issue with the FDA was, though, that we have thousands of patients worth of information on one infusion. But that's something that they gave us a pathway through in our first /phase 2 trial, and that's fine. But the interest there is that in many of these cases, when you have an inflammation that's that strong, there needs to be multiple infusions, and that's generally what I speak about in my talks. Is that if you're going to treat regenerative medicine, it's not going to happen in 2 weeks. It's not going to happen in 4 weeks. It took 9 months for you to gestate. Why would you expect regenerative medicine to happen any sooner? So many of the things we're talking about have to be delayed over time with an understanding of the decay time of messenger RNA, or the half-life, if you will. But what we do know is that nobody is coming out of COVID whole, and that is worse as you get older. But very few people, even people that feel like they had no symptoms, really feel like they did before. So a question that's coming up to me a lot lately is, should you have exosome treatment around your COVID-19 immunotherapy, your vaccine. And I want to speak to that first. So the Moderna vaccine was shown to be 94% effective at preventing symptomatic COVID-19. That was measured starting 14 days after the second dose. So the way we think about viral infection is that you're infected on day 0. By day 7, you should have an immune reaction ramping up. And by day, say, 7 to 10, you're really creating an anti-immune response or an anti-vaccine response or antiviral response. We know it's between days 7 and 10. At that point, you are supposed to have an anti-inflammatory response by the body to stop that response because your T cells, B cells have pretty much killed that pathogen by that point, but you can't have an ongoing inflammation. And what's interesting is that when you think about chronic fatigue, what I believe chronic fatigue is, I locked myself up for 3 weeks to read about chronic fatigue once, was that we have specificities for certain antigens, and everybody's is different because there's this random rearrangement of these genes called JK genes, and you can have a different T cell receptor based on who you are, and it is absolutely random. So some people can react correctly to a vaccine, and some people don't. Those that don't, and those that have these very strong responses are called super antigen reactors. So you have a T cell receptor, which is very large. It's not 9 amino acids long, but there are 9 amino acids that are very specific for an antigen that is presented in MHC class 1. If you have 9 antigens on the other side that are similar, 9 amino acids, and you recognize 9 out of 9 amino acids, say, so let's pretend, take your left hand, that is the Moderna vaccine, the peptide that it produces. In your right hand, you have your TCR receptor. If you brought your hands together, you have 5 fingers. If all 5 out of 5 fingers were complementary to the amino acids on that antigen, on the Moderna vaccine, you would have what is called a super antigen reaction. And that's not what we want. That's not how the immune system works. But in 0.01% of the population, that's what happens. That's why some people die from the vaccine. They die because we don't have a system in place to stop that inflammation after it starts. But in general, and again, I'm talking about 9, but we have 5 fingers. In general, if you took away 2 or 3 of your fingers, you would have what is expected of an antigen response for a vaccine from your immune system. The funny part about it is that the word is promiscuous. You're supposed to have a promiscuous interaction with the antigen. So that should happen between 7 and 10 days, and your T cells start ramping up, your CD8 T cells, your CD4 T cells. Those CD8 T cells start killing any cell that they see that is presenting viral antigen. And then by day 14, this should be petering off, and your regulatory T cells, which you've heard me speak about often, should be cutting off their immune response. Therefore, if you receive a vaccine for COVID, your response should be finished by day 14. And in fact, that is why Moderna, if you reread this slide, that's why they started measuring COVID-19 response from day 14, because that is what is known. So I absolutely do not suggest that you apply exosomes as soon as you know that somebody has COVID. And if you're worried about whether or not they're labeled to this infection, I wouldn't treat them beforehand. I did not treat my mother with exosomes during COVID. I did not treat myself with exosomes during my entire COVID ordeal. However, if I had started to have any kind of pathology or difficulty breathing or symptoms that were stronger than normal, and it's difficult to gauge because with any other flu or virus you had histamine release that caused a runny nose, that caused you to sneeze, and get a fever. I didn't have any of that. That's the scary part to me if you want to think about this as a manufactured pathogen. But it's the truth. The issue is when you're in the ICU, you're a doc, and you have multiple things, hydroxychloroquine, exosomes, you throw everything at the bus. What you want is immunity to COVID. So if somebody has COVID, allow them to have COVID. It's not fun. It certainly was painful to me. I had very painful muscle aches like I've never had before, but they resolved with 2 Tylenol. So isn't that better than potentially adding an immunomodulatory exosome to it? It's too powerful, and I try to say this all the time. Our exosomes are too powerful. When people say, well, this person has more exosomes, well, I don't want more exosomes. First of all, you're manipulating the way the machine works to tell you that there are more exosomes. I can very easily tell you I have 10 billion versus 1 billion particles per ml just by changing the amplitude of the light. That's not what's important. What's important is gaining an immunity against COVID, and that only happens at day 7 to 10. If somebody is experiencing symptoms at day 14, now you have an issue. That virus is probably cleared by then. You can test them for a PCR test, but actually I have the head of the Indochina Naval Response I actually went to graduate school with. He's on my data safety monitoring board, and he's told me how difficult it is to measure that positivity because, yes, maybe there's some nucleic acids that persist in the blood. That doesn't mean that the person is still capable of transmitting the virus. So by day 14, after you've had your first vaccine dose, you should be okay for whatever you want to do next, which could be exosomes, which may need to be exosomes. If they're having that super antigen response I just told you, you may need to give exosomes at that point to suppress that. But the idea that the vaccine needs to be given on day 0 and 30, that was just to expedite immunity. That's not because it needs to work like that immunologically. And that's why the Biden administration is saying, let's forget about the second administration. Let's just give them all first. Because whether you have that second dose on day 30 or day 60 does not make a difference. Whether you had it on day 90 doesn't make a difference. What we need to do is try to grow some kind of immunity in the population as rapidly as possible and with as many doses as possible. So if somebody is desperate for exosome treatment, you are safe after day 14. However, think about the persistence of the exosome as 2 to 4 weeks. So you wouldn't want to take the second dose of the vaccine 3 days after exosome treatment. You would want to wait 4 weeks again so that they get the biggest bang for the buck that they can get. And this is a very serious disease, and many of us will be vaccinated with 1 infusion of the vaccine, but some of us are going to require 2. Why? Because of what I just told you about the variability of the immune system. If you put your hands back up next to each other, left to right again, if we're falling within the normal range of the reaction that we want, some of us will have 3 fingers that will touch the other fingers, and some of us will have 2. Those with 3 are going to get a stronger response, and they're probably going to be immune to COVID. Those with 2 are going to need a second dose. But how are you supposed to figure that out within the population? It's not possible. So therefore, these guidelines fall in those types of ranges. So I hope that's clear. You wouldn't want to give somebody exosomes within 14 days of getting a vaccine after their 14th day, but don't give it before their 14th day. As I said, I had COVID and I never took exosomes when I produced them. The reason being that if I could tolerate it, I want to create as robust of an immune response as I possibly could. If I had started having trouble breathing, I would have immediately taken exosomes. So I hope that helps. So if you can't schedule an appointment exactly 21 and 28 days, this is the CDC telling you don't worry about the second dose. The second dose can come whenever it comes. So why are we here today? We're discussing hair. And while researching this, I became very interested in alopecia areata. I have actually been speaking to providers about alopecia areata for months now, not years, because I knew that as an autoimmune disease, exosomes would be effective for their treatment. But I never thought about it like I have in the last 3 days. Patients that have had COVID had undergone a severe immune insult. And just in the last few days of discussing this idea of alopecia areata as caused by COVID, every day that I've mentioned it to somebody, somebody has told me that they have a friend or colleague or patient losing their hair. In one case, it was that colleague himself. And he had this very typical circular alopecia areata loss of hair. He didn't point it out until after I told him what my theory was. and he said that he was prescribed corticosteroids. Exactly. That's what's happening. You've had a severe immune insult. And normally, immunosuppressed hair follicle, which doesn't express normal immunogenic MHC, for instance, MHC and MHC, it's an immunoprivileged space. I never thought about the follicle like this. Again, it's preparing for this. Just as the placenta is immunoprivileged, just as the eye is sort of immunoprivileged, the hair follicles are immunoprivileged. They have 3 sheaths, there's 3 layers, and they don't express the major histocompatibility complex, which shows antigens which would cause cells of the immune system to kill them. I was thinking about it, is that we have 2 kinds of immunity. We have thymus-based immunity, which happens right after you're born. You have a very thick, robust thymus, but by the time you get to 20, you barely have a thymus anymore. It's difficult to find. The way a thymus works is it expresses all of the antigens of the body such that the body can become tolerized to those antigens, even though it's in the thymus. So it'll see an antigen from the foot. And it won't react in the foot later on because it saw it in the thymus. But that's right after you're born. So you're able to see those things. That's called a local immunity. But you also have mechanisms of diffuse immunity because not every antigen can be shown in the thymus. So a T cell might go to the gut, see something that looks foreign, but it's bathed in anti-inflammatory factors, so it becomes tolerized to that antigen. When you think about hair, hair is really a strange organ or particle, whatever you want to call it. You can't imagine those proteins, keratin or melanin, being expressed in the thymus. So there was not an ability for the body to become tolerized in the thymus. So that's why the hair needs this immunoprivileged status. But if it loses it, the body is certainly going to come after it and destroy it. And that is very clearly what is happening in Alopecia areata and what is happening now in COVID-related telogen effluvium. People's hair is falling out. Not only do they have this problem with their lungs, with their muscles, and I really do hear this every day, but now I'm finding that everyone I ask, and it's only been a few days, is losing their hair. And as I started to research it, you can see these publications here. This is a real occurrence. Many patients that were going to come to you for fatigue or other types of COVID-related problems are also going to be losing their hair. So now you understand why they're losing their hair and understand what's going on. They've created an autoimmune condition. I always like to think about the fact that I became allergic to opioids when I was older because I had a surgery. And they gave me opioids for the pain, and I took them. And because I was inflamed, my body created antibodies towards opioids, so I can no longer take them. That's how our bodies work. And that's why, for instance, we get rheumatoid arthritis as we get older. We lose the regulatory aspects of our immune system, and then we have trouble. Well, COVID just sped that up for us by quite a bit. So it's something to keep in mind. This is an example of what can happen. There's a very rapid thinning, and as opposed to alopecia areata, this telogen effluvium is the cells were in stasis, or the shafts were in stasis, and because of COVID, there's been sort of a shock to the system. The hair starts falling out. Now, nobody knows that this is permanent. This may resolve after 1 to 2 years. But the fact that the patients come to you with other physical impediments is kind of interesting, and it's just manifesting in the hair. So it's characterized by hair shedding 2 to 3 months after stressor. So the colleague of mine that was in the lab 2 days ago, he came in and he said, "How are you doing?" I said, "I'm very excited because I have this correlation of COVID to hair loss." And that's when he pointed out the circle of hair he'd lost. He was 2 to 3 months after. So this is all happening in real time. I'm sure you were all awake 20 hours a day like I was when COVID started. We're seeing this and we're learning very rapidly. So there's a loss of about 20% of hairs. There's a hair thinning. They get very nervous because when they take a shower, they have hair falling out. And let them know that this might be the reason. So what are your options? In this talk, I'm going to talk a lot about other things you can do. You're physicians, you're artists. I work on one specific thing. I'm an immunologist, and I work on exosomes. But you have other methods to treat these patients. You have corticosteroids, as were injected into my friend. You have minoxidil. You have finasteride, which is only given to males because it can cause, but there it can cause erectile dysfunction. It can also cause teratomas in women, so you don't want to give finasteride to women. All of these drugs are tools. Some of them have side effects. In my experience, exosomes don't have side effects as long as you don't have another pathogen on board. If you have Lyme disease or if you have an ongoing bacterial infection, exosomes will cause a Herxheimer reaction and you will see some kind of inflammation, but that also tells you something. But in general, exosomes are safe. Remember, we drink exosomes in milk. Those exosomes have cow proteins in them. They seem to work. So, they're natural versus corticosteroids, which are not really, but do suppress other parts of the system that we might not want to suppress. So can we use exosomes? Well, several factors have been found to be important. We have sequenced our exosomes ad nauseum, and we know that we have a number of proteins that are useful for both growth and anti-inflammatory function. Why? Because they're young exosomes. They're derived from placentas, which is the youngest ethical cell line I can get. And they share some things with cells that are in growth or fetal cells, and not really embryonic. But this would be a good time to mention that as far as the parlance is concerned for cells, you have embryonic stem cells, which are within the first blastulus sort of stage. You have fetal stem cells, which is up until the day this moment that they're born. As soon as they're born, cells from a baby or a placenta or umbilical cord, that's an adult cell. So let's be very clear about that. Okay. So hair follicles go through anagen, telogen, catagen. Anagen is growth, catagen is retraction, and this is normal. It's very interesting to look at the growth factors that are expressed during these times, and they give us many, many different targets. By seeing the types of the depth of research that's been done on each one of these phases, I'm absolutely certain that hair loss is going to be resolved within the next 10 years. It's just too obvious. There's too much research. There's too much we can do to solve this. So what is alopecia areata? It's a non-scarring, and that's important, inflammatory hair loss disease. So what we're looking here is at histopathology, and it's very telling. You can see in the area of the bulb, you can see what's called here a swarm of bees infiltrate of T cells. All of those little purple cells, you should not see that. That's called Huck staining. Those T cells would not normally be here, and this area right here is an area of immunoprivilege. So why are these cells here? Well, because something has upset the system. Some large type of inflammation, i.e. COVID, has occurred and allowed these T cells to infiltrate. What's more interesting, or as interesting, is the fact that we have these cells of the innate immune system that are also there, mast cells, eosinophils. These are regulated by different types of factors. And let's discuss that as we continue. So we've got antigen, catagen, telogen. AA, which is alopecia areata, as we'll say from now on, is marked by a loss of immune privilege. So that means a couple of things. That means that the markers on the surface of the cells in the area are not there. Those markers, which I'm calling MHC class 1, major histocompatibility complex, those generally display those 5/9 figures that I was talking about. If they're not there, then T cells can't recognize them. That's normally good, but if they become upregulated because of inflammation, which is what inflammation does, inflammation differentiates, inflammation causes the expression of MHC, then now the cells of your immune system can recognize it, even if especially if they've never seen it before. But what is also interesting is that research has been done to show that when alopecia areata is occurring, you start to lose the proteins that keep the immunostasis. So if we look here, these are upregulated inflammatory proteins. These are downregulated anti-inflammatory proteins. So we look, what's downregulated? IL-10. What's downregulated? Fast ligand. If you ran into fast ligand and you were a T cell, you would apoptose right away. What's downregulated? IDO. That causes T regulatory expression. What's downregulated? Red, IK, LK. What else is downregulated? TGF betas, all the TGF betas. So everything that we normally think of as anti-inflammatory is downregulated. All of these proteins, even this one, are expressed in our placental exosomes, which I find very interesting. I also find interesting the T cell subsets that are occurring in these types of areas. So I showed you before, this was a swarm of bees. I know that these are T cells, but I don't know what kind of T cells these are. So knowing that tells you a lot of what targets you should go after. So going deeper into this paper, again, that's Zhang 2013 if you're interested. There are far more mast cells and eosinophils here than I would have expected. This is like an asthma. This is an allergy. And so as a physician, think about whatever you could normally use to treat that. And what I'm trying to say in everything I do now is all I'm interested in is the outcome. I'm not interested in specifically whether it's exosomes, because I can harp on that ad nauseum, or amniotic fluid, or dexamethasone or methotrexate. All of these things together is what are going to cure the patient, and that is your job. My job is simply as a PhD to explain to you the immunology of what my part is. But these things need to be used in concert, and all of these approaches are FDA approved over there. Right? They're just not things you want to use long term because of side effects that you should be aware of. But they certainly could help in small doses at the beginning, afterwards to maintain, and should be thought about. So we're looking at that bulb again. And over here on the right, we see CD4 versus CD8. CD8 are killer T cells. CD4 are messenger T cells. They'll tell your B cells to upregulate antibody formation. They'll tell macrophages to become inflammatory. And they will secrete cytokines that make CD8 T cells grow more rapidly. But we do know a lot about the type of inflammation by the numbers of CD4 versus CD8 T cell. If you had purely viral infection, you would have a much higher number of CD8 T cells. If you had sort of a parasitic infection, you'd have much higher CD4 T cells. So what we're trying to do, and if you've listened to any of my talks before, is my goal in life and what I studied in my graduate school career was the conversion of inflammatory CD4 T cells to regulatory CD4 T cells. And we have those T cells for a reason. If I mentioned before chronic fatigue, if you get infected with Epstein-Barr virus and you don't have a regulatory T cell that has a T cell receptor specific for Epstein-Barr virus, your Epstein-Barr virus is going to be cleared by your body within 7 to 10 days, like I told you, but the inflammation is not going to stop. And that's why patients come in with chronic fatigue. And that's why in many cases in my career, I've so easily helped them, was because they had an immune system that was not normal. They were the 0.01% of the population that had a super antigen T cell or receptor for Epstein-Barr virus. And what I mean by that is you either have it or you don't. These people have regulatory T cells that couldn't respond to Epstein-Barr virus. If you go in there and provide some other kind of anti-inflammatory milieu, you can stop that Epstein-Barr virus response and allow them to return to normal. But if somebody doesn't come and do that for them, they stay in a cytokine cascade that doesn't stop. And that is the scary part of it. And the chronic fatigue patients that I've treated have resolved in 2 days. I mean, it's almost like a miracle. And you never see anything like this. But it's very simple if you think of it from that point of view. And if you look at the literature, Epstein-Barr virus is one of these things that causes that. But any virus can cause that. If you have COVID and you resolve COVID because you have CD8 T cell receptors that are specific for COVID, but you don't have regulatory T cells that are specific for COVID, you're going to maintain an inflammation that is going to make you lose your hair, that's going to give you chronic fatigue. So you have to come in and do something else. You have to give them a corticosteroid, you have to give them an exosome, you have to stop that response. or you have to give them an exosome that's going to convert their inflammatory CD4s to anti-inflammatory CD4s. And in fact, you have to trick the immune system. So this is a graph. I'm going to give you a second to look at this. This is the standard of care type treatment of alopecia areata. And no part of this antagonizes. This should all be part of a treatment plan. But these are all small molecule based. Well done. We have excimer laser. I just want to make a comment about ozone really quick while we're here. So ozone is not FDA approved. Extracorporeal photophoresis is. It's been used for 60 years. You can purchase a plasmapheresis device and use that in your clinic legally, FDA approved. The thing is, nobody knows about it, but the mechanism of ozone and extracorporeal photophoresis are exactly the same. The idea is to cause necrotic cell death of cells from the patient, return them to the patient's body. The patient's body recognizes that necrotic cell death, thinks that there's a subtype of inflammation going on that needs to be resolved, then it upregulates regulatory T cells, which immunosuppresses the rest of the body, which gets you the outcome that you're looking for with that ozone treatment. So when you say ozone is anti-inflammatory, it is not at all anti-inflammatory. It is 100% inflammatory. It's just that the response from the body when there is no real infection or pathogen ends up being anti-inflammatory, and you get the outcome that you're looking for. So think about that carefully. If all you're looking for is that anti-inflammatory result, maybe you should think about extracorporeal photophoresis if you want to fall within the guidelines of the FDA. If you can get ozone approved by the FDA, even better. I'm not against either one. I think they're both great, but they're both following the same thought process. So can we use exosomes to treat alopecia areata? Well, I've spoken a lot about inflammation in my past talks. I pulled this out of my Boston Biolife presentation from April 2019. These are the types of papers that I read when I was in graduate school that I really care about. It's the induction of, or the suppression of graft versus host disease using regulatory T cells. And there's 1,000 methods to do that. And guess what? Extracorporeal photophoresis is one way to do that. Upregulating or throwing a patient's regulatory T cells out of their body is also another way to do it. Using IL-2 antibodies to try to upregulate CD4, CD25 positive is also possible. It is not just an entire field. It's probably half of immunology. I mean, this is not a little thing I'm showing you here. People at the University of Minnesota have 8 R01 grants looking at this at the same time. It's really well known, but that's what's happening, and that's what we want to achieve. And if you upregulate regulatory T cells, you will suppress graft versus host disease, alopecia areata, and any other autoimmune disease that you can think of. So we have 2 issues with hair, and especially AA right now. We need to upregulate hair growth, and we need to suppress inflammation. We have 40 employees working at Kimera now. Many of them are involved in the messenger RNA sequencing of our exosomes and our other exosome products. And so when I go through papers like this, I say, do we have this messenger RNA, this one, or this one? Well, I can see that in the current exosome, we have red LK, we have fast ligand, we have SNX1, and we have HGF. Each one of these has been shown in the literature to cause growth. Now, again, I just said shown in the literature. We're a laboratory, and I am producing in vitro models in order to be able to prove these things, as well as in vivo. But this is what I know right now. That in literature, I can show that we have these cause hair growth. What I also know is that this is a video of the resolution of a wound in a horse with exosomes. Now, the reason I put this here is not for wound healing, it's for the growth of the hair. As this wound resolves, the hair grows. And that's a key point. Again, when you see this, I don't know exactly why that wound is resolving, but I do know that the hair is regrowing. So we do know if we have the factors to instantiate hair growth, and we see the hair growing, and we see that there's no inflammation or infection, well, that's pretty good right there. That's what we're looking for. And I really appreciate shooting this the way that it was shot in such that we can see such clear outcomes. So these are some outcomes that we've had from exosomes, and I'm showing them rather rapidly because each one of these could be one of two things. It can be alopecia areata, and it can be androgenic alopecia. This is Dr. Halas, who's also speaking. But again, I'm a scientist, and I try to speak to you about reasons for the occurrences of outcomes, not outcomes or what the patient's status was. If you tell me that that patient was androgenic versus areata, how am I supposed to determine what that outcome was? If I really wanted to say that, I would have done many different types of immunological tests prior to treatment of the patient. So, androgenic alopecia, what is the cost of a head of hair? So there are different treatments for alopecia areata, androgenic areata, but androgenic alopecia. But what I found very scary in researching for today was that one peer-reviewed paper said, while preparing for this presentation, the paper said the primary purpose for hair was to attract a mate, which I thought was extremely strange to read in an academic paper. I've thought about hair my whole life, not as far as a human is concerned, but as far as a platypus or a polar bear is concerned. And I just find it strange for anyone to say that hair, the purpose of hair is to attract a mate. But I haven't even thought about it. Certainly, to many people, that is the purpose of hair, and it's a very important topic. And that's why, in preparing for this, I've become more interested and plan to do a better job going forward. But we have options right now for androgenic alopecia. You know that this is genetically associated. So if we could gene sequence every patient and look at exactly what the deficit is, we could fix it. We do now have drugs called finasteride, minoxidil. These things have side effects. The side effects of exosomes are sort of the side effects that anybody might want in the age of a patient who's losing their hair. So there's more to discuss there, because what I want is the outcome. If the patient has a mixture of minoxidil, finasteride, and exosomes and gets the outcome that they're looking for, then fine. Maybe one can counteract the other. But really, we're not going to keep patients in the use of regenerative medicine if they don't get outcomes. And one issue with stem cell therapy when I started was that many people were upset at the fact that they felt they were duped. In treating many patients in concert with my physician partners, I encountered that 2 or 3 times. And then later I started to realize the reason that they felt they were duped is because that their cells were inadequate, which is why we've arrived at placental exosomes. But it is sad from their point of view for them to say stem cell therapy doesn't work, for them to go tell their friends stem cell therapy doesn't work. And, it puts us back. When a laboratory goes out and says they're selling exosomes and then contaminates 6 people in Nebraska, it sets me back. So, it is just the way of the world. So here's a paper that Dr. Halas is on that talks a lot about exosomes for the use of androgenic alopecia. I believe that these are Kimera's exosomes that are used in this paper. Of course, it's difficult to prove that type of thing these days, but you can take that for what it's worth. So we are developing hair and neuron-specific combination of exosomes and patented nucleic acids to target both inflammation and regeneration. But in the case of areata, to me, it's a clear-cut autoimmune disease, and about 20% of patients with areata have some other type of autoimmune disease that can also be focused on or treated. And so it's a systemic treatment. And so I'm sort of pleading to you as physicians, should I put it locally, should I put it systemically? It is absolutely both. And if I can show you that X protein is downregulated in the local environment of the follicle, and I know that I have that in the exosome, then fine, that's where the exosome belongs. But if you can downregulate inflammation in the entire body of the patient with, say, Humira, and I'm not suggesting Humira, I'm just trying to present an example, with an FDA-approved drug, then you're going to get the outcome that you're looking for, and then you will have repeat business. So that really is the goal. So Right now, exosomes in our hands are carriers of anti-inflammatory cytokines, messenger RNA, and microRNA, which is becoming obviously more and more important. When we characterize lot to lot of our product, we do it mostly based on the microRNA. They're very small nucleotides, but they can be very powerful. But that is where the field is going. You can see it right now by the Maduro vaccine. So the Maduro vaccine is very exciting to me because what is the Maduro vaccine? It's really just an exosome. It's a messenger RNA encapsulated by a liposome, and it's being used to rid, hopefully, our population of this COVID. And that is what we've been doing all along, what I've been trying to build myself up to. So we have biochemists on staff that are working on just Moderna-type vaccines. In many cases, we need what the exosome has. So if you just think about the world as lipid vesicles, about 100 nanometers to 300 nanometers wide for the next coming 10 years, that's what every therapeutic is going to be. And I really believe the pharmaceutical companies are understanding this. So in the case of alopecia areata, our goal is the suppression of inflammation. We want to take an M1 macrophage and convert it to an M2. And this is if you've listened to me speak before, you've seen this 1,000 times. This is the first growth factor panel I did. It's protein-based. I did this in 2017, which is as long as I've been speaking at Boston Biolife. And the first thing I said when reading this, I said, this is like tea leaves. I can see that amniotic fluid is temporarily anti-inflammatory. And I can see that the exosomes are permanently anti-inflammatory. I am seeing this so forcefully lately, it's almost comical. One of the things we do to assay the different batches and lots of our exosomes is we put it in an assay where we excite a macrophage with a bacteria, and we see how much TNF-alpha, which is an inflammatory cytokine, comes out. We try to assay that by ELISA. And when we do that, we see that when we apply our Exoglow exosome, there's not as much TNF-alpha. That's great. That's a nice assay, but that's not the be-all, end-all assay. This is absolutely what I should have seen, but we need to go much, much farther. In this assay, which was recently done, we took our Exoglow and we put it in the presence of microglial cells, which are macrophages. And we looked at how many hours it took for the exosomes to be taken up. Because in this assay, what I'm afraid of is, if you look here, I told you we're assaying TNF-alpha, right? Well, you see here in our amniotic fluid, there is a lot of TNF receptor 1 and TNF receptor 2. Not much in the exosomes. So if we do a TNF assay, where we put in amniotic fluid, then that TNF receptor is probably going to bind the TNF-alpha in that supernatant, and so our assay might not be able to read it. So we might think that we're suppressing inflammation from that macrophage simply because the TNF-alpha was bound up by the receptor of the amniotic fluid. Now, this was something that I said on stage in 2017, but had not done the assay for. I had not done the work. I've now done the work, and it's turning out just as I said. So here, if we're going to say that the amniotic, so if the amniotic fluid and the exosomes are both anti-inflammatory, how am I to know if I really turned the cell off or if I just bound up the TNF-alpha in the supernatant? And so my assay tells me that both are anti-inflammatory, which is what it's telling me right now. That assay is telling me that amniotic fluid and exosomes are both anti-inflammatory. But are we really stopping inflammation at its core? And my goal is inflammation at its core. And if you listen to any of the protocols that we're talking about right now, the reason that we still make amniotic fluid is because it is very useful. Organocell is amniotic fluid. We sell amniotic fluid. Amniotic fluid is a cheap and easy way to stop inflammation in preparation for larger goals that you have. In this case, if we're going to say that we have an exosome and not just growth factors in solution, we need to prove that cells, in this case macrophages, microglia, take up the exosome. And you can only take up exosome if it's a lipid vesicle. So here we apply a DNA stain to these macrophage cells. We then add the exoglow to the milieu, and we can see that 1 hour later, the exosomes are actually entering the cells. And 4 hours, 8 hours, we see that they're taken up more. This is a merge of those two images. But we have proof here that the exosomes are being taken up. I can tell you that we're working on a neural-specific exosome. If this were a neuron you were looking at, and I put our exoglow versus the new neural exosome in the same dish, one of them is going to be taken up more rapidly. And that has a lot to do with which one we're going to choose to treat those types of diseases in the future. So this is a wonderful assay, and I'm very happy that we got that done. The current definition of an exosome by ISEV, the International Society of Extracellular Vesicles, who is the equivalent to the International Society of Cell Therapy, who regulated mesenchymal stem cells 5 years ago and still does. There are tetraspanin proteins on the surface of exosomes generally that you can use to sort of figure out how many exosomes you have. Those markers are called CD63, CD81, CD9. Now that's general, but at least it's a starting point. And that's really what the FDA, God bless their souls, that's what they're releasing things on right now, is that. There's a lot more to an exosome than that, but you can't, when you create something, the FDA didn't, so you have to bring them up to speed. But 63, 81, and 9 are not a bad place to start. So a few years ago, I showed you these images from our ONI microscope, and this is the first light-based image of an exosome that clear in the world. And this is the size of a virus now, mind you. This is actually smaller than COVID, if you can believe that. But we have this microscope, which you can see it. Trust me, don't just buy the microscope because it's not the microscope, it's the user of the microscope that gets it to work. But you can see here that we have these tetraspanin markers, CD81, CD63, on that one exosome. So by that light-based version, we can see that we have an exosome. So my staff, I've been doing this since 2014. We were the first people to sell exosomes on the market. But the people I have working with me now weren't around at that time. So because the market advertises everybody has an exosome now, and in fact, I said that amniotic fluid has exosomes years ago. Since that point, every tissue product now has an exosome. My staff, I'm not sure if they believe me anymore. They want to know what we're competing against. And I repeatedly say two things. I don't want to characterize their exosome for them because I spent a lot of money on this equipment to characterize our exosome. If I characterize their exosome, I'm basically giving them free data. So I refused. But about 2 months ago, I finally gave in and said, all right, let's get everybody's product and let's look at it. This is what amniotic fluid looks like when you get it. It's white and it's filled with vernix. It's milky. It causes amniotic fluid embolisms. It kills mothers that get it into their blood. And I had no interest in taking this wax and putting it on these two machines. The one on the left is $150,000, one on the right is $400,000. We create a very pure exosome in saline that I'm okay touching that equipment. But I am not okay with what you see dribbling from his mouth touching that equipment. I couldn't stand the constant din of my employees asking me to do that. So we went ahead and started looking at everybody's product. Now you'll see no names of other products here, but you will see certain things. We're looking at CD81. CD81 is one of those tetraspanins I just spoke to you about. CD81. Now here we're seeing it with light. This was only accomplishable in the last year. The way it has been done for the last 50 years is by looking at by Western blot. Here we have an antibody to CD81, and if we can see it, then we assume that we have CD81, which is an exosome surface marker. Lane 2, we can see our product. We see CD81. That's great. We further see CD81 here. When we look at competitor A, we see this is the same amount, 100 microliters of each product. We're not doing anything or concentrating it. We see a vague band, and here we also see a vague band. In line 5 here, this is a research grade standard that is used for scientific research around the world for an exosome, which has CD81. And there you see it. We didn't tell the standard company to put CD81 on their exosome. That's just what the standard company knows. We also see it on our exosome, and it's there. If you look at the whole medium before we started growing our cells, there's no CD81. So that's a start. When you look at this one, I had said that amniotic fluid had exosomes in it, but I had read that in the literature. Again, I was afraid to put the baby dribble on my expensive equipment. So we just ran this. We ran our amniotic fluid, and we did, in fact, find CD81. That means there are exosomes in it. We ran our mesenchymal stem cell exosome product, lots of CD81. None of the products on the market seem to have any or CD1 or a lot of CD1. And in fact, this one right here was inflammatory in one of our assays. Again, I'm not telling you who these people are. I'm just telling you that after years of not wanting to do this and thinking that we're exosome competitors, there's no CD81. So here is the next step for Kimera. What we're doing right now is we are taking patented nucleic acids and we're adding it to what we know our exosomes can already do and down-regulating inflammation in different types of models. We are doing rat models of this, we're doing in vitro models, and what I think is most important here is if you look at this, I'm not telling you what microRNA this is, but this microRNA alone in an assay with an inflammatory macrophage can shut down inflammation to this extent. Now this is step one. There are 2 or 3 other targets that you might want to work on. You can couple this with exosomes. By the end of the day, what I would like to see is that this band does not occur versus one such as the in control. And that is the Maduro vaccine. That is what is happening right now with the Maduro vaccine. And that's what we're trying to progress to. So I've shown you this before, but I'd like to show it to you again. When you provide a murine model of mouse exosomes IV, they traffic to the spleen immediately. What's interesting, though, is that they also traffic to the brain. And we've read this in the literature, but it's nice to see our exosomes doing the same thing. And this one, the luckiest mistake that's ever happened, that in each one of these mice, the exosomes immediately traffic to the spleen, but this guy either has arthritis or a bladder infection, and all of the exosomes traffic there. So our next hurdle is in the rats that we're actually studying now for neurological disease, which are going to have much better resolution than these tiny mice. How much of these exosomes go to the brain? And that's pretty exciting to me. And how do we quantify it? Do we draw a gate here such that we say that, okay, 5% went without any intervention, but if we provide some intervention, like focused ultrasound, there'll be 20%. So those are the next steps there. So that's the end of my talk. I'm going back to the guy that started it all. The first You'll Seeing is Believing episode from Garrett from Halloween of 2014 when I applied it to his face. And that's where we ended up now. So thanks for joining us. I'd like to invite you. I think this hair topic is really solvable and very exciting. So as I said at the beginning of the talk, we'll be doing a hands-on discussion of this in Los Angeles at the end of March and the second week of April in Miami. My name is Duncan Ross. I'm the founder and CEO of Kimera Labs, and thank you very much for joining us.
Thank you very much, Duncan. It is always a very insightful presentation, and we appreciate the level that you've gone to analyze and look at this. And really, the commitment that you have from an equipment standpoint is extremely impressive. I think that you're really the only one out there that's characterizing these technologies and these different delivery systems. And I appreciate how you have a broad view of their application. And obviously, tying it back to the coronavirus and COVID, I think is something every healthcare provider is interested in. So thank you so much for this. We look forward to future presentations from you. So our next presentation is by Misti Barnes, and she'll be discussing the pathophysiology of trichology and scalp health. Misti's Celestia's hair follicle rejuvenation treatments have been acknowledged by regenerative medicine and functional medicine doctors and industry leaders. Her groundbreaking technologies and techniques have been used to train professionals across the U.S. So, Misti, thank you so much for coming and being part of our group today. We look forward to your presentation on trichology and scalp health.
Hi, this is Misti Barnes from Celestia's, and I'm happy to be here with you today to talk about scalp health and hair loss from a unique perspective, I believe. I wanted to share a little bit about myself with you. I began my career providing treatments for post-surgical scarring, and specifically with acne scars in burn patients. I was trained by one of the developers of microneedling who worked with burn survivors along with Dr. Richard Grossman of the Grossman Burn Center. My focus was treating scarring, sun damage, acne scars, and ultimately I began developing protocols for hair loss while working with a prominent cosmetic surgeon. I participated in clinical trials, eventually using bone marrow stem cells for a leading hair loss lab in Irvine, and then I was asked to train physicians in the technique that I had developed, which now is known as Celestia. After years of testing and treating patients, I presented my research and findings here at Boston Biolife Regenerative Medicine, and then I began looking at other alternatives during this time for bone marrow. I was starting to look at biomimetic options, including plant-derived peptides, and that's where the genesis of the business began and my research. I then teamed up with a leading biochemist and with a background in hair science. He came from Aveda and L'Oreal. He holds 40 hair loss patents, and together we developed an at-home kit to supplement the protocol that we developed. So this is the genesis of Celestia's Hair Rejuvenation. It's a patent-pending, non-invasive treatment that uses plant-based peptides and growth factors to target hair follicles. Studies of Celestia's have provided very consistent and predictable results with both males and females. My years in the beauty industry as a health and marketing expert and as well as a service provider has given me an insight into patient motivations, what they want, and the practical application of those treatments. These experiences, along with the recent advances in scientific knowledge, led me to Celestia to fill that gap between the hair loss patient expectations and the desires and the existing treatment options. So recently, a study asked 1,407 people whether they'd rather have more friends, more hair, or more money. And you guessed it, 60% chose more hair. So, although science has led us to artificial intelligence and driverless cars, the options for hair loss still are not advanced. Mostly people go for surgery, FUT, FUE, hair pieces, extensions, and while some of them are more reliable than others, the number of options are still very limited. So most of us know about PRP, which involves spinning the blood using the enriched cells, platelet-rich plasma, and injecting that into the scalp with medical needles. It also contains many proteins that stimulate natural hair growth. While PRP contains a lot of cytokines and growth factors and can be used along with placental, amniotic, or adipose stem cells, as well as bone marrow to get results, my experience to date has been that it's a shotgun approach. Because we can't control what is in the PRP and select those biosignals, the ones that are most favorable to restore hair growth, it is not as effective as it could be. Specifically as internally, very effective internally, but for hair growth, it just really hasn't shown to be as effective. Also, we can get trauma from the injections causing shock loss. Exosomes are another option with PRP cells, which uses placental exosomes, tiny packets of substances that contain RNA and proteins that bud off of the cells and carry those messages, the messaging contents to other cells. Those vesicles contain more growth and signaling factors than platelets, but it's a very new approach and more research is being done. I believe that we can trigger the follicles caused by toxicity, inflammation, a lack of circulation, hormones, and many of the obvious like DHT. I believe that we can do this without going very deep into the dermal layer, which often causes inflammation, by targeting those follicles with specific peptides and microneedling. For instance, I moved over from using human bone marrow to using plant-based peptides primarily because of the stability and the consistency within my own practice. So there is a wealth of observational data on specific dermatological conditions such as dandruff and seborrheic dermatitis, psoriasis, and atopic dermatitis, providing evidence for a role of a healthy scalp. While every current treatment for hair loss has been ignoring one very key component, whether it's Rogaine, Finasteride, surgery, PRP, none address the problem of calcification and fibrosis, two of the most chronic progressive elements that are involved in hair loss. The cascade that leads to hair loss is very complex, and my goal is to shed light on only one of these key components that are often overlooked when it comes to treating and managing hair loss conditions. So look at some of the main causes. We know that DHT, fibrosis and calcification, toxicity and free radicals, a lack of circulation at the follicle base, Our lifestyle, medications, hormones, diet, microbiome, oxidative stress, and hormones are many of the causes for hair loss. But most hair loss solutions only deal with symptoms. Whether it's topical, surgical, or medical medication, few address the main contributor to hair loss. Now, in this presentation, I'm focusing here on fibrosis and calcification. Today, we're going to talk specifically about that role of calcification and fibrosis as a downstream cascade that follows inflammation, which leads to the characteristic androgenic alopecia, AGA hair loss. So if we look at the balding sites in patients, the skin feels thicker, it's less pliable, it's less elastic. Causally, we see a shininess, and the more advanced the hair loss, the skin also tends to look more swollen, and that's due to the overproduction of collagen. It's not just too much collagen, it's disorganized collagen, cross-hatchings, which lead to imperfect healing and scar tissue. Studies show that males with AGA or pattern baldness have 4 times the amount of collagen than men with no hair loss. That disorganized collagen is fibrosis. And this disorganized overaccumulation leads to perifollicular fibrosis. We can learn a little bit from scleroderma. In scleroderma, the body starts to overproduce collagen in the lungs, hands, and the scalp. The results in those same visual symptoms in balding scalps, which is tighter, thicker, shinier looking skin, is similar. And then there's swelling. And with scleroderma, those who develop it on the scalp eventually experience hair loss. So we know that excess collagen and fibrosis occur before hair loss starts, and they accumulate, and then the hair loss occurs. Fibrotic tissue has lower blood flow. It's restricted in the thinning area of the scalp, which also scleroderma have poor circulation in the extremities, the head, hands, feet. So it's no surprise that scleroderma sufferers have poor circulation in their extremities. Blood carries oxygen to tissues. So if tissues have lower blood flow, they have lower oxygen levels. Low tissue oxygen is known as hypoxia. The studies confirm balding scalps are hypoxic. In one study, men's balding regions had 60% oxygen levels of non-balding areas. Men with no hair loss had oxygen levels nearly all the same across their entire scalp. So excess collagen fibrosis decreases the blood flow and oxygen choking out our hair follicles, leading to hair loss. The other key component that's been noted in the scalp is calcification. Not only are the blood vessels that indirectly support the follicles in the lower layer of the scalp, not only have they become calcified, the calcium builds up in those places where it doesn't usually appear, like the coronary arteries or the brain. Dr. Fred Holzel and AMA published a connection between calcification, restricted blood flow, and hair loss some 70 years ago. He studied the brains of cadavers and discovered there was prominent calcification. So researchers have long believed that older people have more calcification and that it was quite normal part of aging. But we know that calcification does not have to increase with age. It can be rampant in young people and absent in older ones. Interestingly enough, calcification is not necessarily caused by a calcium-rich diet. So the question is, does calcification cause fibrosis? Not likely. While they may occur in the same areas, they're likely independent of one another. And in relation back to scleroderma, some patients also suffer from soft tissue calcifications, and others just suffer from an overproduction of collagen. So calcification does not have to happen before fibrosis and vice versa. So let's talk about what triggers both. First, when we look at men are most likely to develop arterial calcification and calcified arterial lesions, twice as likely as women. Researchers have long linked it to androgens such as DHT, dihydrotestosterone. Most doctors agree that DHT causes hair loss, but no one actually knows how DHT causes hair loss. If the DHT triggers calcification and fibrosis, this explains how DHT causes loss. What we need to know is, do the androgens like DHT precede arterial calcification and fibrosis? And the research is mixed. On one hand, we see women and men who take androgens, steroids, significantly increase their risk of arterial calcification. But studies have shown that increased androgens don't cause calcification. In fact, they protect against calcification. Androgens alone do not cause calcification. There's another variable that is leading to calcification. Since DHT is the main androgen caused with pattern hair loss, and we know that it alone doesn't cause calcification and fibrosis, what it is the DHT plus an increase in androgen receptors, as well as an imbalance of calcification regulators. So a bit about DHT, androgen receptors, and calcification regulators. If we know that androgens alone don't cause the calcification and that in the body, androgens, they must be reacting with another variable to cause calcification and fibrosis. In 2016, we learned the first one was androgen receptors, the place inside the cell where the testosterone attaches itself. And without it, the DHT does not bind to the cell. It's right here. Interestingly enough, DHT and androgen receptors also increase fibrosis in heart cells. So the receptors aren't always active, but they typically turn on in the presence of DHT or testosterone, and they turn off when they're not present. So here's the tricky part. The increased androgen receptors are not the other variable. We know this because of the DHT paradox. Increased DHT encourages hair loss in the scalp, but it encourages hair growth in the face and the body. So in the hairy, facial, and body tissues, calcification and fibrosis do not occur because in our bodies and face, increased DHT encourages hair growth. That's the opposite of what's happening on the scalp. In the body and the face, when the DHT increases, the androgen receptors must not increase. Otherwise, the body and facial tissues would also calcify and hair wouldn't grow. So it turns out that both balding scalps and hair-bearing body and facial tissues have increased DHT and increased androgen receptors. So something is protecting our body and face from fibrosis and calcification, or something is causing both to happen in our scalps, both calcification and fibrosis. The truth is we don't know exactly what that something is, but 99% of research abides by the DHT argument and genetics, which is making the hair follicles more sensitive to DHT. And for unknown reasons, that DHT accumulates in the scalp and causes hair loss. There really have been no studies exploring the connection between DHT and calcification, but the connection seems to warrant more research. However, in 2016, cardiovascular researchers did find that an increase in androgen and androgen receptors are needed to cause calcification, not just one, but both. So here we have it. Increased androgen receptors and imbalanced calcification regulators, promoters, or suppressors equals calcification and fibrosis. I'm going to cut this short a bit by looking at the studies, and a great thanks to Rob English of Perfect Hair Health, whose vast research I've used, and say that if we inject regular mice with DHT, they develop calcification. If we inject DHT into mice who can't produce androgen receptors, we have no calcification. When we look at mice with androgen receptors who receive DHT, their bodies turn off the proteins that encourage calcium buildup, and they turn off the proteins that usually suppress calcium buildup. That result is calcified arteries. This is really important because surrounding our bodies and facial hair, we do not develop that same calcification or fibrosis that we see in balding regions of the scalp. But the same is not true for our scalp hair. This suggests one thing. There's another variable in play, and that is likely an imbalance of the calcification regulators, the proteins that regulate whether or not our body accumulates or releases calcium. So the conclusion here is that we need these three factors in order for calcification to occur. Increased DHT, increased androgen receptors, and an imbalance of calcification regulators. If our tissue activates too many inducers and too few inhibitors, calcification will accumulate. That explains why the DHT encourages hair loss in the scalp but growth in the body and face. When we look at the causes of increasing DHT, androgen receptors, and imbalanced calcification regulators, we see the main likely cause is chronic inflammation and hormonal imbalances. So chronic inflammation is not healthy. And amongst many other diseases, we can see that it leads to scarring and fibrosis. Increased DHT is not just found in balding scalps, it's also found in inflamed body tissues. There is evidence that DHT helps regulate inflammation and in some cases is anti-inflammatory. So increased DHT is part of the inflammatory process, and DHT binds to tissues after inflammation occurs. And in balding regions, if DHT is chronically elevated, our scalps are probably chronically inflamed. Chronic inflammation is the gun, the DHT, the androgen receptor calcification regulator imbalance is the trigger, and the gun that fires is the calcification and fibrosis, the hormonal imbalance. So the testosterone-estrogen ratio imbalance, which has been linked in women with thinning hair and in younger balding men, elevated estrogen levels are common. Evidence shows that the imbalance in the TE ratio occurs before calcification and fibrosis. And in fact, our TE ratio may actually control which calcification regulators our bodies activate. So if our TE ratio is imbalanced, then we're at a higher risk of calcification. This explains why the imbalances in the TE ratio are strongly associated with heart disease. In men, lower levels of testosterone, they're associated with higher rates of calcification and stroke. Low T men have nearly twofold the increase in morbidity, and they also suffer from higher arterial stiffness, fibrosis. In women, low estrogen levels are associated with higher arterial calcification. Women with polycystic ovary syndrome and high testosterone also have a higher rate of calcification, and the same is true for women receiving testosterone injections after menopause. This is the time when the estrogen levels begin to plummet. So what we want to do is look at this as the cascade that's occurring prior to hair loss. We have to look at things like diet, lifestyle, microbiome, as well as the scalp environment. We cannot separate out the health of the system from the hair health. And you can see from this flow chart how it impacts the hair follicle over time. So chronic inflammation and a hormonal imbalance encourage calcification and fibrosis in the body and the scalp. Diet plays a critical role in both of these factors. Our lifestyle choices are major contributors to poor thyroid function, hormonal imbalance, and the chronic systemic inflammation that precedes the hair loss. Anything that interferes with the ability to generate sufficient quantities of energy interferes with the repair and renewal process of cells, leading to atrophy, infirmity, and the complete loss of functioning tissues and organs. A lack of energy has a ripple effect throughout the entire organism. As cells form tissues, tissues form organs, and organs form organisms. Our gut flora makes up at least as many cells as we have as humans. The influence of gene expression helps us absorb vitamins and minerals and constitutes 70% of our entire immune system. We can focus our attention on optimizing gut health and microbiome, and doing so prevent the chronic inflammation that contributes to hair loss. And our scalp environment affects the natural growth and retention of hair. Pre-emergent hair can be negatively affected by oxidative stress that occurs with an unhealthy scalp. Oxidative stress has been widely discussed in relation to the skin and aging, but the focus has been placed on its role in impacting scalp health, growth, and condition. But little focus has been placed on its role in impacting scalp health, growth, and condition. We must continue to research how it impacts the scalp environment. Bottom line is we're losing our hair because we are no longer carrying out the lives that our bodies were designed to live. We've deviated far from our biological and evolutionary expectations that our endocrine systems are failing us. So the goal of this presentation is to discuss how we can target the chronic inflammation that is occurring and ultimately leading to the imbalanced calcification and fibrosis that's occurring, which ultimately lead to a loss of blood flow, oxygen, and hair loss. Microneedling was originally developed to rejuvenate the skin and reduce the visibility of scars and abnormal pigmentation. In skin treatment, improvement occurs because the body's natural healing response to trauma, even minor superficial trauma. And aside from the minor trauma created, the numerous shallow perforations greatly enhance the penetration of the products applied to the skin after treatment. In the case of hair restoration, we're interested in that trauma as being helpful in 1, reducing fibrosis and scarring, 2, increasing localized blood flow, and 3, enhancing the penetration of the biosignals. So microneedling not only creates a localized trauma, it produces thousands of microchannels that allow us for great enhanced penetration of the topically applied substances. In 2014, a study confirmed that micro-wounding's benefits on hair regrowth with AGA using 1.5 millimeters on humans. Every patient cited hair improvement of grades 2 and 3 on a 7-point scale in just 6 months' time. So we know that microneedling alone, in some cases, can be beneficial for a hair loss patient without any other treatment. That's very uncommon, but it can happen. As evidenced by wounding, regrowth, hair, and improvement in skin scars, if we acutely injure those areas already ridden with calcification and fibrosis, the healing process seems to resolve much of that calcification and fibrosis. When we injure a tissue, our bodies generate an acute inflammatory response that allows the body to repair the injury. If the injury is too severe, like a deep cut, our bodies will repair the wound imperfectly and the results in scar tissue. However, if the injury is minor, like the acute inflammation from microneedling, our bodies will better control the healing process and achieve perfect wound healing. So, in this instance, what we're looking at is the mechanical stimulation that is targeting what DHT blockers, PRP, or surgery cannot target: calcification and fibrosis. I believe that if we can trigger those follicles and target the things like toxicity, inflammation, a lack of circulation, and deal with the issues, the outside issues, hormones, diet, lifestyle, this can be resolved with the help of a mechanical stimulation like microneedling in combination. So if we target these follicles with specific growth factors, peptides, to target the specific issues, we can achieve very impressive results with plant-based options. And in my personal practice, as opposed to, say, using bone marrow, placental amniotic, or adipose stem cell tissues. I personally moved over from human bone marrow to biomimetic in my own practice, primarily for stability and consistency. So in reversing calcification, the best method we've seen to reduce that is through acute injury, and which is why I use the microneedling. So throughout my years of research and study, I found a technique that allows for short-term injury with low inflammatory time with a needling device that I'll tell you more about later. Microneedling not only increases the localized trauma, it produces the channels that we just discussed, allowing for better penetration. I do want to discuss the effect that the microneedling has on the calcification regulation. Acute wounding appears to downregulate the calcification inducers, like transforming growth factors, while upregulating calcification inhibitors that promote scarless healing. This acute inflammation generation produces the right balance of calcification regulators. We need all three factors as discussed: increased androgens, increased androgen receptors, and imbalanced calcification regulators for pattern hair loss to occur. Acute wounding addresses the third variable and reduces some of the calcification and fibrotic tissue also already present. Acute wounding also promotes angiogenesis, the formation of new blood vessels and capillary networks that provide oxygen and nutrients to our follicles. So if we create new blood vessels around thinning follicles, these new blood vessels provide those thinning follicles with blood and oxygen. And by reestablishing that blood flow and oxygen, we can encourage hair thickening and even regrowth. And in addition to these mechanisms, mechanical stimulation upregulates genes associated with hair regrowth and downregulates genes associated with hair loss. Mechanical stimulation targets as I mentioned before, mechanical stimulation targets what DHT blockers and surgery can't: calcification and fibrosis. So this research has led me to the development of Celestria's hair rejuvenation, so while we're treating the patient in office, we must be doing something at home. Because even with these other treatments, continually triggering and through mechanical stimulation, continually triggering the follicle is key. Celestrias can be used as a standalone protocol or in conjunction with PRP or exosome therapies. Most of us are familiar with peptides, and with PRP, you've got your scaffolding in place, and you can use the Celestrias protocol to support and enhance the results that you're already getting. It can be used prior to other protocols or in conjunction with them to help increase the rate of results. And specifically with our home kit, which is key to that continual ATP production, we find that the home kit is really helps support what's happening in office. So there are many ways in which we can use the combination. We'll discuss that in a moment. But I wanted to share with you kind of the results of the multiple years of R&D. Originally, the first study was with peptides, strictly peptides, GHK. The effectiveness of the treatment was dependent upon the treatment timing, penetration, depth, needle size, and frequency. Then year 2 was peptides and bone marrow stem cell study. I'm happy to share this information with you if you reach out. Year 3 was peptides and bone marrow stem cell cytokines, 60 clients in total. And then in year 4 was the formulation of Celestria's hair follicle rejuvenation. We started with 55 clients, developed the formulation. The treatments were every 2 weeks. Then we moved to 4 weeks, And these were consistently highly predictable results. Each client received between 6 and 8 treatments over 3 to 4 months using the at-home protocols with the serum and lipid replenisher, and this increased the success rate. Scientists have long thought that people with hair loss have a depletion of hair follicles and follicle stem cells, of those necessary to grow hair. We now know the bald people have the same number of follicle stem cells as those with hair. They've just gone dormant, and it's the inability of these stem cells in the scalp to develop into the type of cells that hair follicles make, and that might be the underlying cause of male pattern baldness. Scientists know these follicle stem cells need biosignals from which within the skin to grow hair. Topical growth factors and peptides may be more efficacious since they address the physiologic biosignaling that's in control of the hair follicle function, which is a more natural and physiologic approach. When growth factors are combined with microneedling, which supports the follicle by stimulating the epidermal stem cells in the hair follicle bulge, shifting the follicles into the anagen phase, we have an effective combination. We can trigger these follicles caused by toxic inflammation, a lack of circulation, hormones, without using more inflammatory options and targeting the follicles with specific growth factors. So I'm going to share with you some of the studies. Our serums contain a proprietary blend of 9 of the most powerful peptide combinations. Here's a sampling of some of the trials and one of the combination of the ingredients. And if you're interested in reviewing other trials, just email me and I'll provide that to you. The trial included 20 volunteers aged 21 to 62 for 150 days, male and female. Here are some of the results. What I'm going to discuss now is the blend of peptides that I found to be the most successful in the treatment of hair loss. So the key growth factors and proteins, as listed here, are formulated to reduce inflammation, target the dermal papilla layer, and stimulate the hair follicle stem cells with 20 amino acids vital to the hair follicle health, tripeptides, including red clover, rosemary, saw palmetto, and biotin, in order to support hair health, reduce atrophy and aging, and oxidants and anti-inflammatories to help prevent free radical damage. It also contains a combination of ingredients that includes, but it is not limited to, Antigen and Versapil. So first, Nicotiana benthamine. This is a 5-alpha reductase inhibitor that prevents the conversion of DHT from testosterone. FGF7, dermopapilla signaler, to instruct the hair germ cells to proliferate and initiate a new growth cycle. IGF-1, insulin growth factor 1, which strengthens the hair and stimulates hair follicles to produce a stronger hair shaft. KGF stimulates capillaries, endothelial cell migration, proliferation, and differentiation. EGF to help with scalp blood circulation and revitalizing follicles. Biotinol GHK to increase the production of cellular binding elements to block the DHT. So also fights follicle aging, strengthens hair from the roots, and prevents loss. VEGF, stimulation and facilitation of nutrients, feeding the hair follicle through the process of induced angiogenesis. GSH, a DNA protectant to reduce lipid preoxidation and oxidative stress. And the Vitis vinifera fruit marrow stem culture boosts the turnover of the human follicle dermopapilla cells, nourishes the blood vessels, regulates the thickness and the growth, and increases the hair lifespan. So this is the combination that we have been used. Our products and serums are manufactured in an FDA-approved and compliant lab. I'm going to share with you a little bit about the protocol that we're using. This combines an in-office treatment protocol. It takes under an hour in the office, and it includes a home kit for the patient, which takes about 10 minutes, 3 to 4 nights a week. This comprehensive program, we also include supplementation direction as well as comprehensive review of the patient's health, lifestyle, diet, microbiome, and hormonal conditions. So we'll often, do labs and make recommendations on supplementations. This is part of the protocol that the doctors use in combination or as a standalone treatment option. But what I found is it's very important to look at the whole of the person and to be able to get an analysis and a purview of what's actually happening with the body. We treat people with multiple conditions, and at the end of the day, what's happening within the body, and as we've discussed earlier, hair loss is not the diagnosis. Hair loss is the symptom, and we need to treat the symptoms. So I'm going to run through our professional kit. It is a disposable kit. It doesn't require labs or blood draws, so it reduces the concern of contamination. This is what the professional protocol in-office kit comes with. The serum pre-packaged, shipped directly to your office for each treatment and can be thrown away immediately. Also comes with a needle, a disposable casing, a coupler, and a tip. In a moment, I'll go over our training, which includes 5 in-office kits as well as the proprietary microneedling machine. The Celestia's home kit is that combination of products that it's a consumer version of the peptides that we're actually using in the in-office protocol. What I discovered early on was that there's no such thing as a one and done. Even if it's once a month, every 4 weeks, we must be continuing to trigger the follicles. This is a chronic problem. It's progressive and chronic. And so the more we, treat it as such, the better our results will be. So I tell my patients to look at it as exercise. It's something they're always going to need to do. And under times of stress, the hair may start getting, we might start seeing loss again. And that's when I tell them to just get back on their program. But the more they look at it as exercise, because we want to continue to trigger those follicles, the more successful they'll be. So compliance is really important in this treatment. Here you see the serum. It comes also with a scalp stamp, a leave-in conditioner, and a scalp replenisher. These are all designed to support the growth of the hair. Also for penetration, the stamp is used for penetration. The scalp replenisher is to cut inflammation and also to support hair in the anagen phase. And then the leave-in conditioner, which is great for those with telogen effluvium or hair, a lot of fallout. It helps prolong the life's hair cycle. So this routine makes it easy and part of a grooming regimen. So most patients find it pretty easy to do. It also supports the results of what we're getting in office. So I'm going to talk about who's a good client for Celestria's. Obviously, healthy adults, all ages and ethnicities. We can use it as a pre-transplant or a post-transplant to enhance the results and to diminish the fibrosis. 6 months to a year post-chemo. Males and females with AGA, thyroid, menopausal, hormonal-driven hair loss are successful. And patients receiving PRP, exosome therapy, can use it to enhance their results. Ideally, when it comes to hair loss, the younger the better. The better and the quicker we can get to resolving the problem, the better. But I have treated patients in their 70s, and we've gotten nice results, into 20-30% rate, which is pretty nice for someone with an aged person with many health issues, and I'll share that with you in a moment. But I've now treated over 600 patients with Celestria's, some with some pretty incredibly compromised systems, including HIV, blood disorders, and many with multiple autoimmune disorders. And we're still in the 90% success rate. Again, so if we consider that success, new growth between 20 to 40% vellus hair. It can work on the frontal lobe of the scalp, although those areas tend to be more difficult due to a lack of circulation and a lack of fat in the tissue. But we do find it just takes a little bit longer. I'm going to show you now real patients and some of their issues. As I just discussed, multiple health issues. This is a client in his 70s in the beginning. In the middle, multiple surgeries. I find the fibrosis obvious in calcification with a patient who's had multiple surgeries, FUE, FUT. And this is an example, the middle one, of a patient after just one treatment. You can see that inflammation in the center of the head is starting to reduce. I just pulled that up as a second, the second treatment, so you could see how well he's doing. The third, this patient has a blood disorder. This response actually took 9 months. I treated her once, and then I said to her, I didn't think that I could help her. She called me 9 months later, and this is where she was. So she only had one treatment. It's not typical, not something I'm claiming would happen, but there are those success stories. So here are some more of the typical cases I see. Traction alopecia. You can see the chronic inflammation here and collagen dysfunction in the first one. Then, I do see in California a lot of my patients that are inhaling marijuana, which, I have a couple of articles if you're interested in look. I'll send those to you on the relationship between THC in inhalation and hair loss, and the fact that it causes people to rely more on cortisol. It affects the whole system and gets it out of whack. Anyway, and then we have here just a typical early AGA patient, another client with thyroid issues, just nicely filling in. And this is just typical androgenic alopecia of a patient in their 60s. So we can get really nice results. And then here are just some other. This one was interesting to me because she has a parasite in her system and multiple autoimmune issues, another autoimmune, and this is just strictly AGA. Wanted to show this because this shows you how we really, looking obviously at things like dermatitis, which I brought up earlier, and that's a whole separate discussion of how to identify dermatological conditions. Sometimes we find that with the protocol, the scalp, we just need a few weeks, a few treatments for the scalp to heal and to come out of its inflammatory state in order to really be receptive and start growing. But as you can see, this is a nice example. Really want to go into a little bit of what patients are looking for that I found over time. They want something effective, they want something non-invasive and painless, they want something that's low risk, and they're looking for non-toxic, drug-free solutions. They don't want downtime. They want it to be easy. Basically, they want no commitment and everything at ease and not a lot of compliance. So we've tried to keep those things in mind and make it as easy on the patient as possible. They also now, with COVID, of course, want to be able to rely on telemedicine. And while that's not the ideal scenario, I definitely need to see people in the office and want them to have labs and be really analyzed. I know that there are people that need a solution and they can't get to the doctor's office now. So through that, we can offer the home kit. They also want a good price, of course, as do all providers. A provider wants high ROI, if something effective and easy to administer, not time consuming, no labs, and low risk for cross-contamination, painless for their patients, long-term revenue, and a low-cost investment. So what we're offering here with Celestia is it's non-invasive, painless, low risk, drug-free, effective results, no labs, it's disposable, very little time, no downtime for the patient, easy to maintain for the patient, a high return on your investment, easy to administer, low risk for cross-contamination, long-term revenue, low investment cost, and can improve the results of the biologics and surgery, of other options that you may be using, PRP, etc. So if you are currently not using a full biologic option, peptides are a fantastic option. If you are, Celestia can be used to augment that. It can be used post and pre-neograft surgery to prep a patient and to maintain the results, also to reduce the fibrosis inevitable in surgery and some of the calcification that may be occurring at the follicle base. So this is the next level of improving patient results, which is obviously what they all want. So if we look at the financial benefits to a office or practice, it's a great opportunity to expand the market to include hair loss patients with a non-invasive, non-drug option. It's an immediate, positive, long-term, consistent ROI from in-office and retail sales, and it doesn't require the doctor's time, costly labs, or blood draws. We provide the training and the support and licensed materials, as well as online, digital, and social media assets to help a physician or a practice to begin promotion, as well as offering your practice on our website under the Contact Us, Find a Provider page. We also offer guidance on how to promote and market the service with our training. We do offer additional ongoing support, which would include marketing and advertising strategies, including newsletter, blogs, and SEO support for monthly fee. This is an easy yet intensive training, even for non-medical staff, and it is dependent upon state laws. Some states allow estheticians or mid-level professionals to treat patients without medical supervision. That's, of course, dependent upon your state. But our easy startup includes 5 office kits, machine, marketing materials, social media support, before and after photos, easy training, and as I mentioned, they can be administered by RN, PAs, MAs, and non-medical staff in some instances. Here's an example of the actual protocol. So that gives you an idea of what the protocol looks like. That was a very beginning setup. What we're going to be doing in the treatment is several passes to the point of we're ablating the skin, but we're not going very, very deep. As I mentioned earlier, we don't need to go incredibly deep. We don't need to go beyond. We're barely going at 1.0 in terms of depth, 1.5, 1.0 to 1.5 over a series of passes. So that was just an example of a demo there. So, here are the numbers, a little bit more information about how you can benefit by offering this treatment. And at $400 per treatment with just one patient a day of a home kit, the office can earn $161,250 net. That's just if a patient came in once and never returned and bought a kit, which is, I've never had that happen. There's always that one chance it will, but it's actually a really nice ROI. It's 300 to 400% per treatment. So you can bring this protocol to your office at a cost of $3,500 for on-site training. It includes the didactic and hands-on training, proprietary microneedling machine, 5 office kits, 5 home maintenance kits, before and after images, and practice building marketing assets, as well as retail products at wholesale pricing. For a short time, we're also offering live trainings in the New Hampshire, Texas, and Florida area at a special price of $2,500. If you're interested in that, and we do have fewer spots because of all that's going on, but if you'd like to book a spot, give me a call or reach out and we can let you know what the options are for that. So here are a few testimonials from doctors.
This particular technique is different than, say, microneedling or conventional microneedling or injection of platelet-rich plasma into the scalp. This technique is much less painful. We just did a procedure within the last few minutes. We just finished, and this fellow's pain was out of a 10. He only had a pain of 1 or maybe 2. The other thing that is, and this is important for lots of people, is that it's not nearly as expensive.
Because it works. And if they do have a concern about their hair loss, I think it could be life-changing. I'm in the natural healthcare business. The people that come to me are not really wanting too invasive of a medical procedure. They come to me because they don't want surgery. They come to me because they don't want drugs. So the fact that it's a natural plant-derived product is just a natural fit for my patients.
From an employer standpoint, I think the infectious disease risk is much less. And for a patient perspective, it's more economical and less painful. It does not involve a blood draw, which is stressful for them.
Thank you for allowing me to share this information today. As I've mentioned, hair loss is the symptom, not the diagnosis, and it's a complex problem. There are no overnight solutions or easy answers. After years of research, I've come to the conclusion that the more compliant the patient is, the better, and the better the results are. But it is a whole person concern. We have to look at the whole of the person. If you have any questions, would like information on training, or would like to read any of the articles I discussed, or look at the clinical trials, I'm happy to send those your way. My number is 213-536-9339, or you can reach out to us at info@Celestia. Have a great day.
Misti, that was a very informative talk, and I think that you did a good job of outlining the personal precision approach that these different technologies have, as well as sharing your wealth of experience with us. So we appreciate that perspective, and I think it dovetailed in very nicely with what Dr. Ross had said. And okay, we're going to get started again, and thank you all for staying with us. We have a good audience today listening to these fantastic presentations, and really thank the excellence of our speakers for the commitment that they have. This isn't easy to do. And also Michael Cameron, who's the mechanic behind the scenes putting all this together. So our next presentation is Dr. Yael Halas, and Dr. Halas will be discussing her use of exosomes in clinical practice and how she looks at them for prevention and restoration. And Dr. Halas is recognized as one of the best facial plastic surgeons in New York City. and she uses a combination of techniques, and she's been trained in thousands of physicians in her surgical and non-surgical techniques in lasers, injectables, doing rhinoplasty and others. So Dr. Halas, thank you so much for sharing your experience and pioneering into the role of exosomes in hair restoration.
Hello, everyone. This is Yael Halas. I'm a board-certified facial plastic surgeon, and I practice in the center of Manhattan in New York City. The bulk of my practice is in aesthetics, facial plastics, and I would say at least a quarter of my practice is dedicated to hair restoration. I'd like to shout out a big thank you to Boston Biolife and, of course, to Duncan Ross and the team at Kimera for inviting me to speak today on the latest advances in the treatment of hair loss, and in particular, how exosomes has been helpful to my practice and, importantly, to my patients. So let's dive into the topic. Hair loss is an enormous issue. Global alopecia is valued at over $7.5 billion in 2015, and that number is just growing and growing. And by far, over 90% of cases is androgenetic alopecia. That is both genetic and hormonal hair loss, which is affecting over 50 million men and 30 million women in the USA alone. Women, you may be surprised to hear, experience a decrease in hair density starting as early as age 20, with the hair diameter generally decreasing after age 30. Noticeable hair thinning in women, they'll start complaining in their 40s. And this general thinning of the entire scalp and also widening of the midline part. So this is not just men. Women too are experiencing this. And this is something that you can offer to both genders. Some statistics, as we were talking about, 35 million men. This is the general pattern that we see in men that everybody is well aware of. Men will be talking about it as early, as early as their late teens and early 20s. But as we get to age 60, 65% of men are going to be noticing. And for women, that is a significant number as well. We know that about over 75% of women over menopause are going to be complaining about hair loss as well. So this is a lot of patients who are going to be complaining about this. Now, for men, we're easily talking about it. It's considered a normal process of aging, but for women, we've been silent on that topic, and we really shouldn't be. The average adult will have 100,000 to 150,000 hairs, and lows up to about 100 hairs per day. And there's some simple tests you should be doing in your office when you come to evaluate a patient. For instance, just the normal pull test, and you should familiarize yourself with some of these basic tests. And you should also familiarize yourself with just the basic follicle growth cycle. Antigen will be an active phase for 2 to 6 years, catagen a transition phase, 2 to 3 weeks, and telogen, which is kind of a very important phase to be aware of because it's the resting phase, which can be 2 to 3 months. And that is going to impact very frequently how your treatments are going to be affecting the hairs, because very frequently a normal hair can just be in a resting phase for 3 months. So keep some of these important facts in the back of your head. And then the most important, in fact, that's been really impacting our patients recently is COVID. If we look right now on the internet at the 2 most searched beauty terms since this pandemic has struck us, first one actually is acne or maskne, because we're all wearing these masks and it's really having an effect on our skin. Number 2 is hair loss. And Duncan is touching on this in his talk today, how much hair loss is affecting all of us. COVID, coronavirus, has been either directly because of immune response or viral response in our bodies, but also because of really, honestly, stress. We know that stress, besides physical stress, but emotional stress, can really cause a tremendous amount of hair loss. And this is why it's become the second most researched beauty term, and everyone is complaining about this pandemic literally causing our hair to fall out. So this is something we really want to know a lot about. So I had already mentioned that androgenetic alopecia is the most common hair loss that we are seeing. There's a bunch of other terms that we should be aware of and where exosomes may be able to make a difference. Some of the most important terms that you need to be aware of and thinking about telogen effluvium is hair loss that can be a temporary hair loss due to a stressful occurrence, be it a physical occurrence or a medication or temporary hair loss or surgical. Traction alopecia is extremely important, and we see this very frequently from too tight braids or hair extensions. Alopecia areata, many of us are aware of, is a hair loss in a patchy area very frequently due to an autoimmune response. Scarring alopecia is something that's particularly for those of us who are doing hair transplants really need to be aware of, because we don't want to be transplanting patients in that area, particularly when it's in the active phase, but once it's in the quiescent phase, they can actually be good hair transplant candidates, but it does need to be differentiated from androgenetic alopecia. But potentially, alopecia areata, for instance, there's been a couple of studies that it's been responsive. Obviously, it's responsive for corticosteroids, but PRP has been written about as a potential treatment successfully. And question arises, could exosomes? Looking at the pathology of androgenetic alopecia, we do know for men the importance of 5-alpha reductase, and that's why we have alopecia. Unclear for women, and really what we've come to understand is the progressive diminution of dermal papillar cells and leading to eventual hair loss. And very frequently with all of these treatments, whether it's PRP, whether it's Propecia, and now we're talking about exosomes, are restoring the hair as it miniaturizes into sort of full-grown hairs, and that is really what is causing quote-unquote regrowth. As I was mentioning earlier, all three phases really can occur simultaneously. One strand can be in anagen, another in telogen, another in catagen. And so we really need to pay attention to the hairs being in these different phases, and respectful that some may just be in that resting phase. Respecting that and looking at it a little bit more frequently, a little more intensely, we can see that we can do things to maybe shift hairs from one cycle to another. 85% of the hairs are actually going to be in that growth phase, and perhaps we can push hairs from one into another cycle, but some are going to be normally 10 to 15% of hairs are going to be in that resting phase, and that is normal. I mentioned already, just in passing, Finasteride, and these are some of the treatments that we have FDA approved and we know exists very well. Minoxidil is another one, brand name Rogaine, and some of the ways that it is effective. Let's jump into one of the kind of more exciting treatments, and one of the reasons why we're here today is exosomes. And one of the things that Duncan already talked about, and I think is important, is donor source is key. Know your donor. Some regulatory concerns, and this is definitely something that I was very concerned about when I was looking into moving from PRP, moving into stem cells, moving into exosomes. Is what were some of the regulatory concerns, and I know some of the other speakers have spoken about that today. None of us want to work with something that has a warning on it. None of us want to work with something that has an FDA warning. And I personally take my patient safety into tremendous consideration, and am comforted by the fact that we have IND now for Kimera, and that it is a very tested product. And Duncan could speak more to this, the difference between 361 and 351, and moving towards having an IRB and having a safely tested product on the market. What do exosomes deliver? So you're getting anti-inflammatory cytokines, androgenic growth factors, and basically the importance of messenger RNA. Messenger RNA has become even more important and more familiar to all of us because that's what's in the COVID vaccine that I personally have taken and I'm grateful for. Growth factors. There are so many growth factors, and this is much more than what you're getting in your doses of PRP. And why it works? Well, we're really trying to, and this is what we're trying to figure out, is the best timing, and when we're seeing this, is to really trigger with our injections a little bit of an inflammation, a little bit of some stimulation then to get some proliferation, and then some remodeling. Exosomes are not small cells. This is not stem cells. I am not harvesting fat, which I have done. I used PRP for many years, and still do use PRP, and then harvest and still do on occasion harvest fat from my patients, and then break it down to nanofat, theoretically, hopefully getting stem cells from that nanofat, and do inject that into the scalp to hopefully stimulate hair growth, and that can be effective. But exosomes are not small cells. What we're really looking for is that messenger RNA, and really hoping to get those growth factors, those billions of or millions or thousands of growth factors, to really help stimulate the effect at our terminal source. Because of the unique stability of exosomes, we are really getting an excellent message delivery. They are frozen without a need for cryopreservatives. They are not rejected by third party, and they are tested for sterility. And I would love to say that this is a result from exosomes, but no. These next few pictures that I'm showing you are hair transplants. And hair transplant is still the gold standard. But what we can do with exosomes is coming, hopefully, maybe tantalizingly, maybe we can get close if we figure out how. So let's talk about how I do it and what I've been doing. So basically, for the past year and a half, I have been injecting exosomes into the scalp, and this is exactly how I do it. I defrost the exosomes, very similar to the COVID vaccine. You have to keep the exosomes vial frozen. This is something that has to be taken extremely seriously. You don't want it to be defrosted for more than a half hour. Because you can lose some viability. So in my office, we defrost exosomes only once the patient has confirmed therapy. We taper the dose depending on the area that we're going to be injected. We draw up the exosomes into 1 milliliter syringes, and we inject using 32 gauge needles. That's the needles that I use for Botox. We do subcutaneous injections about every centimeter squared. And for the scalp, we find that our patients really don't need a ring block. We don't really do any ring anesthesia. We just use Prodox, which is just a name brand for the inhalational nitrous oxide. And that really works well for the vast majority of the patients. In general, I'm using about a 5 milliliter for a full scalp. For a frontal hairline, I can use about a 2 milliliter. I'm going to repeat this again and show you with some examples. An article that I wrote summarizing some of my initial experience, along with colleagues from Toronto, and Boston Biolife will make that article available and give you some information available for those of you who are interested in reading more about the article and our experience, and why we think a little bit more specifically why it works. Okay, some examples, which is what really everybody wants to see, and how I did it and when. So these photos are taken usually about 3 months after, and basically what and how and when and how was I following these patients. So I started injecting in about August a year and a half ago. As I said, I use about 5 milliliters for the scalp. I'm injecting. Some people have larger heads, so I will go up to about 7 milliliters. I do not dilute. I am not injecting with ACell. I do not inject with PRP at the same time. Particularly when I started, I was injecting with only a single treatment of exosomes, and I injected one time and then followed the patient at 1 month, 3 months, 6 months, and then a year later. And I just did a single injection session. This is a 5 millimeter Kimera in a 51-year-old male suffering from androgenetic alopecia, and you can see the results. I will say that the vast majority of patients often said, well, if this is the result that I got from one, can I do more? And a number of the patients have come back, and now we are doing a second treatment to see how they do. With the discussions with the company and also just because of patient interests, we have a couple of arms of investigation going forward, and those include I have, and I will show you some of those patients. I have a number of patients who are getting injected, as I mentioned, for a second round. I have a number of patients who get injected for a second round 3 months apart. I have a number of patients who are getting injected first time with exosomes, then coming back monthly in between with PRP. I have a number of patients who we are doing first time with exosomes, then doing a small focus areas, for instance, maybe 5 ml all over the scalp, and then doing a small focused area with a 2 ml or a 1 ml. What the best answer is, I can't say exactly yet. Here you can see the response on the frontal area. Another 5 ml. We have another, it's gray hair, but you can see how much increased density we have. Scalp, another 5 ml. You can see the improvement in density. Female patient, we have a lot of female patients who've been quite happy. 41-year-old woman. You can see as is common with a lot of female patients, they'll have maintained the frontal hairline, but they will thinning throughout, and you can see the improvement in her density. This patient, like many women, will have frontal hairline, but thinning of the frontal hairline. And what I really like to show here is how much we were able to bring her frontal hairline down. So if you look at how on the right, the density of her frontal hairline, but also how her forehead has basically been shortened. And that is because of, again, all of these, this is a single injection. So really a nice improvement right there. Very similar, a different patient. And you can see a tremendous improvement. This is from a single injection. And when you compare the distance, and it's not even perfectly lined up, but it's just an incredible result. You can see just how much less forehead she has and temples because she had just such an increase. And this is just a focused 2 millimeters to her frontal scalp, one single injection a few months later, and you can see the improvement in her frontal hairline. Another interesting, so this is not 5 millimeters just to her eyebrows. This is 5 millimeters and a couple of very interesting things about our patients. So we did 5 millimeters to her frontal hairline, and we took a little bit to her eyebrows as well. And so I would say maybe less than half a milliliter was actually used for her eyebrows because a lot of it was to her frontal hairline. And this is just after 1 month. She was so excited that we just did, she's so amazed that we just did another 1 ml to her eyebrows. And we've been seeing a lot of really good result with eyebrows. So not something maybe everybody has thought about. There was a recent publication I just wanted to include from my colleague Diane Duncan talking about using from another company, but I just thought it was interesting. Whenever there's a publication talking about exosomes used topically for fractional CO2 in the recovery, showing superior results for acne scars. And we also have been using them topically. And one thing I wanted to point out, I mentioned that I just use sub-Q injections. I do not do needling. I don't have the patients needle or do anything more at home to their scalps. When I do use it for scars, we are doing it with microneedling. I'm not injecting for acne scars. We are microneedling it. We can use it as a glide, or we use it topically, and it is not injection. Here it is, we are microneedling and then applying it essentially topically. So you can see the generalized improvement in his acne scarring. This patient was thrilled. We also used it, and there'll be a close-up, If you can tell, he has a facelift scar running in front of his ear and then also running kind of behind his ear and down his neck. It may not be so impressive in the pictures, but for this gentleman, that facelift scar was extremely troubling to him, as you can imagine, having absolutely no hair with which to camouflage it. So that we could microneedle it and make that scar, one, less hypertrophic. It was kind of really widened, and the texture of it was really quite noticeable. It really was life changing for him. We did a close up to hopefully you could try to appreciate it more. On the left, you could see it was kind of a little bit bubbled up more, and on the right, it was much smoother. This is after only 2 treatments. You can see that it's smoother, it's flatter, it's not as noticeable, and certainly in lighting it was much more noticeable. And for him, this was actually really dramatic. I will go back for a second also and just show that I did a rhinoplasty on him too, and yeah, tip is so much nicer on the right, so that made him happy also. Okay, so very nice, and you can also see on the right his skin texture much improved as well. This was also just a nice result using my 3D camera on the right. But this is a patient that came to us, had a burn, had resurfacing done elsewhere, was extremely traumatized by the experience, and we did microneedling with exosomes, and really such a nice result. So, and then just the future, hair gene and cloning. Patients always ask me, and I always say, every 10 years, we're 10 years closer. And it's true, we are. But unfortunately, every 10 years, we are 10 years closer. So, this is the latest. We have the hairless gene in science in 1998, and there is some interesting human-to-human hair transplant being done actually at my alma mater at Columbia University. Thank you.
Thank you, Dr. Hallis. That was a wonderful presentation, and I think it's important that you, how you were able to illustrate how you not only use exosomes, but you use all the different techniques in your practice and help your patients on an individual basis. So thank you so much for that. We really appreciate the time that you've taken to be part of our group and share your wonderful insights and your experiences. So our next presenter is Dr. Alan J. Bauman. Dr. Bauman is a world-renowned expert in hair restoration. He is the president of the Bauman Hair Hospital. He's a hair transplant surgeon, and Alan literally has been lecturing around the world for over a decade and pioneering many of the new techniques. And he's probably one of the first doctors to incorporate a full spectrum approach to hair restoration, not just transplants, but also all the preventative methods. And Alan brings a tremendous amount of experience and credibility to the field. And Dr. Bauman, thank you so much for taking the time to be with us once again at Boston Biolife. We look forward to your presentation right now.
Hey, thanks, Joe, for that great introduction. Again, I'm Dr. Alan Bauman, board-certified hair restoration physician, coming to you from Boca Raton, Florida. And today we're going to talk about the effective medical management of androgenetic alopecia. And that's what I've dedicated my entire professional career to. And we have a lot of fun doing it, helping people look good and feel great. So if you do want to learn a little bit more about my background, I encourage you to check out the website. So we're just going to jump in today and get going with what we do here at what I would consider the hair hospital, and that's located right here in Boca Raton, Florida. And this gives you a nice overview of how we treat hair loss today. So here at the practice at Bauman Medical, we do everything from hair transplantation and, of course, bioregenerative medicine for hair regrowth like PRP. Of course, a lot of different education and training programs, and you'll learn a little bit more about how to get into the field of hair restoration and how to help your hair loss patients as we go along today. But I wanted to give you a more general overview, because not only are we doing transplantation and these bioregenerative treatments, but also managing the hair loss process. And that's what we'll focus on today, as well as scalp health. We do a lot of research and consulting for businesses large and small around the country and around the world. And we also do non-surgical hair restoration. So patients who are not candidates for hair transplants can use 3D printed cranial prosthetics or hair and scalp prosthetics as well. So pretty interesting stuff here at Bauman Medical. We encourage you to check us out a little bit further if you do have some interest there. Today, we're going to talk a little bit about hair and self-image, of course, the physiology of the hair follicle, hair and scalp health, and an overview on hair restoration treatments and procedures, and then a little bit about next steps. And of course, you can always send me some questions at any time if you have a question about hair loss or hair restoration or anything that's related to those fields. So for the millennia, hair has been a sign of beauty and youth. And maybe this was the first Facebook post, I don't know, but she seems to be checking him out, although he does have a little bald spot there in the crown. But a lot of our friends have told me that beauty and youth should also be accompanied by fertility when it comes to hair. And it's no question that hair is a sign of that for the ages. And when you look good, and you also feel good too. But we also know that hair loss affects a lot of people here in the United States. Probably close to 100 million Americans are suffering from hair loss, about 50 million men and about 30 million women. And so there's a lot of issues going along with the hair loss process, because when it happens to you, it makes you feel bad. You can have fear and anxiety. You can have an impact to your self-esteem and self-confidence. Feel older, less attractive. Many of my patients, especially guys in their younger ages, will avoid social situations or wear a baseball cap to hide their hair loss. And in this day and age, gosh, the internet is packed with information, a lot of misinformation actually, about hair loss and hair restoration, and this can delay treatment. And so patients will doctor shop, they'll search for miracle cures and so forth, even run around to their local physicians, derms, OBGYNs, primary care physicians, anti-aging docs, and get batteries of blood tests to no avail, and really without any effective treatments. So we really want to get a handle on what we can do about the hair loss process. And it all starts with really the coolest organ in the body, right, which is the hair follicle. The pilosebaceous unit, as you can see right here, has a lot of components to it. And that's what makes it such an interesting organ, not just for research. It's got certainly a very, very active metabolic profile, but also you can see the circulation and attachment to nerve endings, its proximity to fatty tissue, as well as its own oil gland, and now we know its own sweat gland, its own muscle. Erection occurs obviously at the level of the hair follicle. So it's a pretty complicated little organ that we've got there. And what I always say is that it's a highly accessible dermal appendage, which is a musculo-neurovascular endocrine mini organ containing a stem cell niche. Capable of degeneration and regeneration, which altogether can elicit a significant emotional response in the owner or the observer. And so for hair fiber production to occur of these 100 to 150,000 scalp follicles that you have right there on your head, what we know is that 84% of them are in a growing phase at any given time, and they're going to produce about a half an inch to a quarter of an inch of hair every month. So that's a lot, about 1,600 inches a day, or about 6 feet of hair per hour is made at the level of the scalp. And it takes a lot of energy to do that. If you try to create a gram of hair, which is about the amount of hair that would fit into, like, say, a Sweet and Low packet, you'd have to do over 10 minutes of vigorous exercise to burn the same amount of energy. So your body commits a lot of energy to making hair, and maybe that's why it's so important in terms of its impact on how we look and how we feel. So if you're not sure what the follicles look like under the scalp, here's a strip of scalp with the hair follicles embedded, and you can see the base of those follicles, the root of the follicle, the dermal papilla, which is embedded deep within the fatty tissue, the fatty layer. And that does tell us a lot maybe about the interaction between fat cells and those types of stem cells and the hair follicle perhaps. But we all know hair follicles turn on and turn off over time. So the anagen, which is the growth phase, is followed by a degeneration phase of catagen and telogen, and then that cycle continues on and on and on over time. As that cycle continues, what I want you to be aware of is that the hair follicle will be producing hair typically for about 2 to 6 years at a time, and then about a 90-day resting phase before it kicks in and grows again. And unfortunately, this can trigger a shed in between the cycle, and so which can also trigger quite a bit of panic, obviously, in a patient who's experiencing it. But 100 to 200 hairs is basically normal. And I've given you this little video here. I hope you can see it, that shows a little bit about really the activity and action that's going on under the scalp at any given time, hair follicles turning on and turning off and so forth. Because sometimes when we look at our scalp, it looks kind of static. But this chronobiology, or this hair follicle cycling, is really under tight control. And we know a lot today about these cycles and what controls them. So there's really two centers within the cell that communicate, or two centers within the hair follicle that communicate between themselves. The bulge of stem cells, which is located about a third of the way down the hair follicle, close to where the erector pili muscle attaches, and the proliferating progenitor cells of the dermal papilla, which is at the base. And so these two cell populations communicate back and forth to control the anagen, catagen, and telogen, or the growth and resting phase, the alternating phases of hair growth over time. And this is very, very tightly controlled cell signaling. This image may look like a alphabet soup if you're not familiar with the different growth factors that are related to hair growth. But the interesting thing is that we know a lot now about these different growth factors and how they turn the follicle on and off. It's kind of like a chemical switch. And we want to be able to hack the follicle. We want to be able to turn the follicle on when we want it to grow thicker, stronger, healthier hair. And a lot of our treatments and therapies are designed to really get into this hair follicle cycling so that we can turn a follicle on and keep it on. The other thing that I want to tell you is that the hair follicle stem cell niche is not just the follicle itself, the bulge and the papilla, but also the surrounding dermal fibroblasts and the adipocytes, and they all play a role. And to take this analogy a little bit further, in order to grow good quality hair and have healthy hair follicles, of course, we want the soil of that forest to be super duper healthy. And we know today that scalp health is a major concern for many, many people out there. If anybody knows what the most popular shampoo is in the world, I'm going to give you a little hint. I'm going to tell you it's Head and Shoulders. And so can you imagine over 100 bottles a minute, or almost 39 million bottles a year of Head and Shoulders is being sold? And why is that happening? It's because people occasionally, probably 50% of people out there, have irritated, inflamed scalps. And so it's not a good environment for healthy hair growth. We want to try to address that, certainly. And here's just some photos that you can see of an unhealthy scalp and the inflammation that kind of goes along with it. This can be accompanied by symptoms like itching, flaking, oily, or even dryness. And we want to try to impact that if we're going to grow good quality hair. A lot of the data and a lot of the research now has kind of been directed at this multifactorial pathophysiology of hair loss, which includes some intrinsic assaults on the hair follicle, for example, like stress and so forth, as well as inflammation that could be happening in and around the area of the hair follicle, as well as environmental assaults, things that we apply from the outside. So as I mentioned before, hair follicle chronobiology is a very tightly regulated process, and inflammation can interrupt this. Repeated exposure to inflammation can actually shut the follicle down permanently, and so we definitely want to try to interrupt any kind of inflammation that's occurring. Damage to the hair follicles can be temporary or can be permanent. We have to interrupt any kind of insult to the follicle at the earliest possible stage. And so, again, a strong concept. There are all of these different insults to the hair follicle which interrupt the cycling. Everything from the natural aging process to heredity, or response to different hormones, our response to cortisol and stress, diet, nutrition, to you name it, things that are happening from the outside like pollution, products and styling and such, which can really shut the follicle down, literally pushing more follicles out of the growing phase and into a resting phase. And that's not good for a healthy head of hair. Just some symptoms here on scalp health. We've got, as I mentioned before, itchy, oily, dry scalp, as well as coarse hair thinning, hair breakage, down to sores and scaling and crusts on the scalp are important signs of poor scalp health. And as I mentioned before, some of the internal factors and external factors, which I would typically go into more detail in some of our more robust training classes, like the hair coach program that we do here at Bauman Medical. But taking a quick step back, male pattern hair loss, androgenetic alopecia, is the most common thing that we treat here at Bauman Medical. 98% of all hair loss is genetic, and we know that it's visible from across the room. If you're a guy, you can see it happening, receding hairline over time and then extending into the crown. And we know that DHT, dihydrotestosterone, which is a derivative of your own natural testosterone, is that trigger which causes the hair follicles to miniaturize over time. And that's important because the mainstay of therapy could be something to block either the the conversion of testosterone to DHT, or the effects of the DHT. Now, in women, it's a little bit more complicated. I'm going to touch on just a few things here. Genetics and heredity obviously play an important role in women, as well as illness and diet, nutrition, stress, hormones, surgery, scalp health, and of course this inflammation that I mentioned before. But we get a different type of pattern in women. We don't often see as much of a hairline change as we do a depletion of density, a diffuse loss of hair quality and quantity, typically in that frontal zone just behind the hairline. Sometimes we can see a receding temple area as well. Other types of hair loss in women that we see in the practice, telogen effluvium, like excessive shedding, as well as traction alopecia, often caused by different hairstyles, braiding, pulling on hair, like from extensions, and then some of the more scarring types of alopecia, like frontal fibrosing and cicatricial, are a little bit outside of the scope of our presentation today, but we do teach that in our training classes. So here's what the female hair loss scale looks like. What I want to bring to your attention is that we can see that 50% of the hair you can lose 50% of the hair before it's noticeable to the naked eye. So even before you get halfway through this scale, you can lose over half of your hair. That's not such a good thing. So 50% of hair loss in that middle zone. If you're looking with the microscope, however, you can see that happening a little bit more clearly. And so these are some of the photos that we take with the dermoscope or hair cam right in a clinic situation here in the office, where we can see those miniaturizing hairs in both men and women, those hair cycles that are being affected, so less density, less caliber hair. And we know why that's occurring. We're trying to figure out, is it the genetics, is it medications, illness, stress, and so forth that's causing those weaker, thinner, wispier hairs over time. And then taking a stab at what are the different types of treatment strategies. What could we do? What are the tools in the toolbox that we would typically use to address the hair loss situation? Of course, it starts with the medical history first and goes along from there. So trying to identify and quantify what those triggers and the risk factors are, taking a look at someone's medical conditions, medications, lifestyle, and so forth, identifying those and trying to reduce those triggers or risk factors in a patient is an important part of treating the hair loss situation. Again, taking a look at the scalp health, we can do that with a variety of different tools and techniques, and then correct that scalp health problem with different approaches such as scalp treatments, or what we call the scalp makeover, even simple changes in hair care regimen, decreasing inflammation, for example, or other issues at the level of the scalp. Decreasing androgen effects, certainly important in men and certain women. We can use pharmaceutical and nutraceutical approaches to do that. Increasing hair follicle function, this is probably the area which we've seen the most improvements over the past few years, trying to address hair follicle function not only with chemicals and pharmaceuticals, but also with nutraceuticals and organics, laser light or red light therapy, regenerative medicine techniques like PRP and PDO grow, the use of bioengineered or recombinant growth factors, stem cell therapy, you name it. But of course, if the follicles are dead and gone, we're going to need to redistribute permanent follicles into those depleted areas. And of course, that's what a hair transplant does, removing hair follicles that are resistant to the hair loss process from the back and the sides of the scalp, and implanting them literally as little as one at a time into the thinning or the balding area using an artistic approach. A quick overview of some of the hair loss risk factors. I'm not going to spend a lot of time on this, but we all know, as I mentioned before, poor scalp health, smoking, as well as exposure to general anesthesia, even high fever and viruses and such can affect hair follicle cycling over time. And this long list of medications, from blood pressure, cholesterol, anxiety, depression, to birth control and thyroid medications and hormone replacement in men and women, can also trigger these effects in the hair follicle. Looking more closely at scalp health and trichology, we have ways to examine the scalp using a variety of different scientific methods, such as scalp pH level, moisture level, sebum level, looking with the microscope under blue light and white light microscopy to identify oxidized sebum, and really getting a complete picture of the health of the scalp non-invasively, and then performing maybe a scalp makeover, which is a diagnostic and therapeutic treatment regimen that delivers treatments dependent upon what we found during that examination and optimizing really the health of the scalp. So scalp makeover is a do good, feel great treatment. You can see here the improvements in the scalp, but it also feels good, feels very relaxing when you have that kind of a treatment done. We have different devices now that are available to help us accomplish this. The hydrofacial and Keravive system is great. You can hear it and see it working right here, right on the scalp to exfoliate as well as to deliver different proteins and peptides, for example. On these two different patients, male and female patients, the hydrodermabrasion technique is exceptionally good, as I said, to deliver different peptides and proteins as well as to exfoliate. Of course, over time, we want to track the hair loss situation, so we'll do a baseline measurement with the hair check trichometer. This is a non-invasive way to get literally measurement of exactly what's happening at the level of the scalp over a course of time. And this is something that we teach at our hair coach program, certainly, and you get actually the kit, a starter kit, which has the hair check device, and you would learn how to use this tool hands-on. And here you can see a way that we would describe and depict hair mass index changes for our patients over time, which can track the response to treatments and also encourage them to maybe be a little bit more compliant with therapy, and also determine if you're doing some kind of an intervention like a PRP treatment, when would you do the next treatment? Because we know that those therapies don't last forever. The effects don't last forever. Digging deeper into treatment strategies, we know a lot about how laser light therapy, nutritionals, and these other types of medications and growth factors work. And so, again, these would be reviewed in more detail in our treatment and training programs. But as an example, low-level laser light therapy is something that can be used at home. This is a non-chemical, non-invasive treatment. Here, this slide is featuring the newest, latest, and greatest turbo laser cap, which is the most powerful, most portable, and fastest treatment time of all physician-prescribed laser therapy devices for hair regrowth, and it is FDA cleared for that. And here you can see some examples of a patient who had some regrowth over the course of a year with laser light therapy. Nutrition and nutraceuticals is something that we also focus in on, teach, and train. Aside from just providing the follicle with good nutrition, obviously we can address the levels of cortisol, inflammation, as well as hormone levels with nutraceutical intervention, and even getting down into the microbiome of the gut and the skin using different probiotics these days, very powerful. Both Viviscal and Nutrafol have some very good peer-reviewed journal articles which support their use in terms of density, diameter, and so forth. And we would review some of those during the Hair Coach program and recommend those routinely to our patients. In terms of pharmaceutical intervention, what you'll see is that we're typically prescribing now not the over-the-counter Rogaine anymore in terms of topical minoxidil, but we would use a compounded minoxidil. It's going to be a lot more gentle on the skin, but powerful on the follicle. It's not going to be a greasy or gooey mixture. And this is something patients would typically use twice a day. So we teach how to instruct our patients to use it correctly and how to get good results with that. Here you can see some before and afters after some periods of time. Usually visible results with these kinds of therapies take at least 6 to 12 months. If you don't remember, the double-blind, randomized, cleverly crossover study performed on Finasteride from years ago. That's the oral anti-DHT medication. You can see it here where they showed a very strong dose-dependent result from oral Finasteride. We do prescribe that a lot in our male patients as well as in our postmenopausal female patients. We would review the chance of sexual side effects as well as the positive effects from oral Finasteride in our patients. You can see here a nice response from oral Finasteride in this patient before and after 10 months. Because of the concern of sexual side effects with Finasteride orally, we sought out a compounder to create a topical Finasteride product, and this is called Finasteride, or Formula 82F, I should say, which contains Finasteride also and minoxidil. And so it has a very positive effect on hair growth, has a low serum concentration of Finasteride when it's being used, and much lower risk of side effects. Not as effective as an oral treatment, but again, a great option to avoid the sexual side effects. If you're concerned about those with the oral medication, you can use this topical. Some of the magic happens, however, though, when you combine these therapies. Patients always want to know what happens if I use this plus that. So here you see some examples of topical medications plus red light therapy, formula 82M minoxidil plus laser. You can see this in these women who are in their 50 years and older. Very powerful effects, non-surgically, obviously. Just medication and laser light therapy alone, with or without nutraceuticals. And so these 3 different women, very impressive effects from a combination of non-invasive treatments. Sometimes you can see the results happening at 3 months. Sometimes it takes a little bit longer. One of the latest and greatest regenerative medicine technologies I'm sure you have already heard about is PRP, platelet-rich plasma. And so that's what we dive into next. We do hands-on training for PRP, really using a bespoke methodology of dual spin process along with the signals and scaffolding to really get a very powerful hair regenerative effect. Of course, as we know, platelets are the workhorse of regenerative medicine used in tissue repair, wound healing, as well as skin rejuvenation, dentistry, spine surgery. Gosh, you name it. There's so many different areas of medicine that are leveraging the growth factors within the platelets. And today we know exactly what those growth factors are doing at the level of the scalp and how they increase proliferation, differentiation, and survival at the level of the hair follicle, and prevent really the apoptosis or cell death that occurs at the level of the dermal papilla. We also know what the correct dose is, or at least we think we know the nice dose relationship between the number of platelets that you can concentrate with these spin procedures and the effect at the level of the scalp or the skin. And here you can see there's a direct relationship between platelet concentration and mesenchymal stem cell migration and proliferation, as well as new blood vessel formation. So 1.5 million platelets per microliter, that would be our target dose of platelets when we do a dual spin process in the office. And those of you who've heard me speak before know that I can go on and on about our dual spin procedure. And but I think it's one of the major breakthroughs that we've learned, really what gets a strong effect at the level of the scalp. So PRP for hair loss is an autologous therapy. It uses a patient's own platelet-derived growth factors as an outpatient procedure over a lunch hour. It's a comfortable, low-risk process. We know how it works, and our results are very well documented. And this is an example here of one of our PRP classes. We're teaching and training how we apply, not only how we create the PRP, but how we apply it correctly. And so spinning the blood down, obviously, with a dual spin process through centrifugation. To get to that 1.5 or more million platelets per microliter, this is how we test it right there at the bedside using a Coulter counter. You can see the baseline platelet count at about 250,000, which is normal, and then our platelet count, which is 1.6 million platelets per microliter. And so what's in your PRP? If you don't know, if you're not getting good quality results with PRP currently in the scalp, then maybe we should be looking at this kind of dosing strategy for the PRP. And these are some of the results that we're getting in patients who are good candidates for the process. So both male patients with some early stage thinning can get some reversal within the year, usually between 6 and 12 months. We've seen some nice improvements in hair quality and quantity. And in our female patients with that widening part line, the thinning and miniaturization in that frontal zone, also in the early stages, can get some great results. We've also seen some interesting results in patients with traction alopecia, where the follicles are still alive, not dead and gone, using PRP in these areas where they've either had damage from braiding or the application of wigs or weaves and such, or even hair extensions. Now, interestingly, in this day and age, a lot of patients have not been able to come into the office due to social distancing and or stay-at-home orders. And so we've developed a new procedure called virtual PRP that they can do at home, utilizing the same combination of different types of treatments that we would use in the office, basically leveraging growth factors and cytokines in an at-home therapy that was through recombinant cell culture conditioned media technology, which I'll mention again in a moment, as well as some nutraceuticals and laser light therapy combination for at-home use. And we do teach how to apply this process in our hair coach program, and you would have access to this combination of therapies for your patients should you decide to learn a little bit more about it. If you're not familiar with the way that this technology works, you need a source of mesenchymal stem cells in the laboratory. So the cell culture is expanded and conditioned in a particular way to get a separation of cells and then a secretion or a secretome product. And then that secretome is then collected and essentially bottled, sterilized and such if need be, and then sent out to physicians to provide to their patients. And so our virtual PRP, our at-home kit, would include laser light therapy, microneedling device like the dermal stamp, as you see here, a liquid stem cell stimulator, which is the growth factors and cytokines, in addition to a stem cell support nutraceutical, which has a lot of good peer-reviewed research behind it. And so this came out of a clinical trial that we did using these growth factors and microneedling in the office, and I'll just show you a couple of results here. Before and after 16 weeks of treatment using this kind of therapy, you can see a rejuvenation of color and quantity of hair in this patient, a 55-year-old female, getting some nice improvements, as well as in some of our male patients. We had about 12 patients in the original clinical trial. Interesting results all the way through, which we would also can present to you during the Hair Coach program. We also obviously have the access to the different types of patient information formats like the videos that tell you how to use these treatments at home. Diving into my favorite form of hair restoration, we're going to talk a little bit about hair transplant surgery. Of course, no presentation from Dr. Bauman is ever complete without a little chit chat on hair transplants. And of course, what we're doing today is not pluggy or painful anymore. It's not going to result in a long linear scar. We don't do it that way anymore. This is the minimally invasive approach of hair transplant surgery, where we're going to take the individual follicles or grafts one at a time from the back of the scalp using a very tiny instrument, usually a 0.8 millimeter rotational tool. And you can see here kind of what the follicles, what the grafts look like, containing sorted here 1, 2, 3, or 4 hairs each in the petri dish there. Here's an up close view of what these follicles look like when they've been extracted from the body. You can see the nice intact structure of the follicular unit, the groups of 1s, 2s, and 3s. And if you look close, you can even see some of the sebaceous glands that come in along with it there as we do the harvesting. Of course, the best thing about this kind of harvesting is it doesn't leave any linear scar tissue. We're only taking a small amount of hair follicles from the back of the scalp at any given time. And so here's a patient 24 hours and 14 days out from the procedure. They can return back to normal, if you will, without a bizarre looking hairstyle. And of course, his results here before and 12 months out from the procedure, you can see the hair regrowth in a natural pattern that gives nice coverage to the scalp, not overstated, wispy, wavy hairline design using artistry and this follicular unit technology. So follicular unit micrografts really is the workhorse of the procedure. We use large sessions of these to create an artistic and natural hairline, and the angle, the orientation, and position of these follicles are critical in order to get the end result, which looks soft and natural. It's a process by which we can recreate density for both male and female patients. About 50% of our patients are women coming through the door these days. And if you notice on the lower right-hand side, sometimes the hair has less to do with what's going on top of the scalp and more to do with what's going on between the lips sometimes. But as I said, when you start to look good, you feel great, and that's the benefit of hair restoration surgery and hair regrowth treatments in general. Here's some more examples of our patients going through the process and regrowing their hair. On the lower left, you'll see this gentleman is about 6 months out from the procedure, getting a little peach fuzz action going on there, and of course, growing all the way through the 1-year result. Now you're looking on the right-hand side, the restoration of the frame of the face. It's a local news anchor who had a procedure done with us. And patients of younger age as well, great candidates for hair transplant surgery. You see a patient like this in his mid-20s, hiding under a baseball cap, trying to get stabilized with medical therapy, then ready for transplantation, and you see the changes there, not only in the hairline, but also here in the temporal point as well. Important to see the improvements and how that changes up the frame of the face. And in female hairline design, women's hair loss can affect the hairline in many ways, shapes, and forms. Women's hairlines have many more variations, and so we can create things like increased temporal density, widow's peak if need be, or not for our female patients. A lot of women do come in, want a lower hairline. So you'll see here on the upper left-hand side, a woman 24 hours out from the procedure, and then a year after with the regrowth in the lower hairline. And on the upper right, you'll see here eyelash transplantation after severe scarring from an injury or accident that we did through our pro bono programs. And on the lower portion of this slide, sideburn restoration after facelift surgery. Very common request here in South Florida because the sideburns are typically ignored, removed, discarded, destroyed by plastic surgeons here and everywhere else. So putting that hair back is important to get a natural look and also to hide the scar tissue. Female patients can do very well, as I mentioned before, with hair transplant surgery. This is after 2 separate sessions of transplants for this young lady here. And for those of you who may know my history, the first person that I met who had severe hair loss was my dad. And you can see his results here from bare to hair after 4 sessions of hair transplant surgery, approximately just under 9,000 grafts. We did his procedure a number of years back, and so of course I'm proud of his result, and so is he. Although he's a little bit more gray these days, he's still got what looks like a good, thick, full, healthy head of hair there. So moving on from the hair transplant surgery, many physicians, I should say, want to know what's next. Well, patients and physicians, they want to know what is the latest and greatest, what's going to happen with stem cells, and can we regrow an unlimited supply of hair potentially for our patients? And I can say, well, it's a definite maybe. We don't know yet, but we're getting closer. So I'll introduce you to Nico, the nude mouse on the left over here. He's born and raised in Japan, and he's got increased self-esteem and self-confidence because he's got a bunch of new living and growing hair from stem cell therapy on the back of his nape of his neck over there. And so he's the envy of all nude mice in his portion of the lab there in Japan. But on a serious note, we are doing this kind of research right here in this country, here in the U.S. A friend of mine, Alexei Tursky, down in near San Diego, California, has been working with induced pluripotential stem cell therapy to try to grow dermal papilla cells combined with epithelial cells, as well as the use of a biological scaffold to create what you see there is a couple of new hairs. And that is the first step in a long journey to create an unlimited supply of hair for balding patients. So long way to go, but he's got a couple of first steps under his belt. So pretty exciting stuff. Congrats to the guys over there in San Diego. They're doing great, and they're going to keep us posted on this amazing technology, and certainly we'll keep all of you posted. But when it comes to treating hair loss, kind of taking a bird's eye view now, it's all about collaboration, and this is what we teach and train. And we've really developed a very profound network here of not only physicians and trichologists, but also cosmetologists and estheticians, everyone from the hair replacement industry to manufacturers and pharmaceutical companies to medical device manufacturers and so forth are really working together to try to help those patients who are struggling with hair loss. And this is something that I encourage you to become a part of. And the best way to become a part of that is to start helping hair loss patients in your own practice or get better at it. To ask any questions at all, you can text that number 561-220-2628 at any time to get a copy of the presentation or to get your questions answered. And I hope this was valuable, at least a nice introduction to hair loss and hair restoration. Thanks so much for your attention today.
Okay, Dr. Bauman, thank you very much for that wonderful presentation. And I think it's important to reiterate why you all do this and why people care, is because you're really trying to make a difference in patients' lives. And Alan, I think you did the best at really thinking about that from that perspective, and whether it's a regenerative medicine treatment or something more far out from a laboratory or some of the basic mechanical things that you recommend, I really think showing that patients can take control of the situation and actually make a difference in their lives is really beneficial and must be very rewarding for you as a clinician. So we're going to do our Q&A session now. Dr. Bauman, you're there, so perhaps do you want to just give us an opening statement as to kind of where you see the future of regenerative medicine and hair loss? And I'm not sure you heard all the presentations, but whatever insight you had, I think would be helpful.
Thanks, Joe. I did get a chance to watch a brief portion of most of the presentations today, and it was great that you put this together. So thanks so much for doing what you do and getting the word out about how fun it is to help patients who are struggling out there with hair loss. some of the exciting things that we've been doing in the practice, as you saw in that last presentation, relate to regenerative medicine. And of course, that's where you and I met at one of your Boston Biolife conferences. That's what it was all about. So we just continue to take it further, and it's a pleasure to be here with my esteemed colleagues who are really on the cutting edge. Thanks for putting it all together. I really appreciate it.
Duncan Ross is really the reason why we're doing this, and I'm just really a mechanic that is able to leverage the assets that we've built from relationships that you've helped us create, and Duncan has helped us create over the years with our in-person meetings. And we find now that people aren't traveling, we get more and more people coming to us for advice, and it's nice when we can have updates. And for all the things the pandemic has brought us that are negative, it's really given us an opportunity to focus on immune health, to focus on personalized precision health more than ever before. And for someone like me, it gives us the ability to churn a lot of content quickly from people like yourself, and clearly Duncan Ross, who's now developing new ways of developing these technologies. So Duncan, maybe you could say a couple words about Kimera.
I haven't had a chance to listen to Dr. Bauman that clearly or precisely before, and that is a lot of information. It's so dense. And I hope this came across in my talk, that I feel a little silly that it is so dense and that I'm just sort of scratching the surface. But I did get really excited in the last couple of days because, again, I don't want to be a jerk, but I didn't know how complicated this issue was. And I didn't know how much literature there was. And there is a lot of literature. And I really think that the answer is right there. We can see it. There's an inner sheath, a middle sheath, an outer sheath. Some of these are there during Antigen, some aren't. Science progresses over time. So if you called it a striated fibroblast in the year 2000, it was probably a mesenchymal stem cell. And that has changed. That understanding has changed. And the growth factors are progressively known. So what I've learned over the last week was really encouraging. We can manipulate these exosomes. Other people, remember, you're standing on the shoulders of giants. Other people have isolated what the problems are, and now I get to come up with a resolution, and I love that. But I know 3% of what Dr. Bauman knows, and it's scary how deep it is. But it is definitely solvable, I think, within the next 10 to 15 years.
Yeah, I think what you're illustrating, though, is sometimes ignorance is bliss. And when you come in from Alan has and you just look purely mechanically at the pathophysiology of the anatomy and the biochemistry of the treatment, such as what you do, and you compare that to your other experiences from immune health, right? I think you start to see the correlations that all the doctors are figuring out. And, you talked about chronic fatigue syndrome, right? And so when you look at the cell danger response, we look at mitochondrial health and all these things that people thought were crazy before, there seem to be these common threads.
Privilege of a hair follicle, it doesn't express MHC class 1. So if you could take an allogeneic hair follicle and put it in there and not express MHC class 1, is that the approach? Or is it the exosomes from those? And the different cell types that are discussed are myriad. There's many, right? So you have to know what the growth factors are, what really the messenger RNA are, and who has done that. Nobody has done that. And it needs to be done and looked at very carefully. But some targets are there, and I'm very excited to approach it. The question in my mind is, do you take one target and use that, or do you take 5 at one time? Do you take these systemic approaches as well as those 5 targets? That's really what complicates my thought process.
Well, that's why we got to do some studies, Duncan. Come on, let's get some product into these patients' scalps, and see what happens.
Right. So as long as we're saying that, so it's people's scalps. In terms of Areata, I have been teaching people that lymph nodes to the upper levels of your head are over here behind your neck, right? That's where inflammation occurs. Inflammation doesn't actually occur where you have a psoriasis, right? It occurs in your lymph node. So I just want to get that across. We have to cause the suppression of inflammation in the node, but we also have to move down and target the inflammation where it's starting. So you have to go to the follicle and you have to go to the node. And I just wanted to let you think about that for a second.
So there's Dr. Hallis with us. Dr. Hallis, do you want to say a few words about the presentations, or do you have anything to contribute to the panel? We're just getting some opening statements before we actually field a couple direct questions that came from the audience.
Hi.
Thank you for a wonderful presentation. I think you did a great job. We tasked these professionals in somewhat of a last minute fashion, and you know, it's very hard for them to drop what they're doing and to try to encapsulate everything they're doing and to put this presentation. So I hope people really appreciate that this isn't trivial. It's not something simple. It comes with great consideration and effort. And we worked late into the night to get Dr. Hallis's talk together, and I think it came out really well. It's almost if you all knew what each other was going to say, which is always the magic when we try to bring people like you all together, because you don't know what each other's going to say. But the beauty of it is that you actually support and complement each other very nicely.
I'm excited, and I'm grateful that you were able to put this together, and I've been excited to be working with exosomes for the past year and a half. Yes, we did work late in the night, and I'm popping in between my office hours, and good to see you, Alan. I feel like I haven't been to an ISHRS meeting in a while, so hopefully we'll all get to be together at some point soon. And I think exosomes is really going to change how a lot of us who do hair, and certainly my colleagues in facial plastics as well, I think there's a lot of use for it, for lasers and skin healing. Certainly going to shift the paradigm away. We've been relying a lot in the past few years on PRP, and I think it's really shifting the paradigm. So very interesting times.
Let me argue something out loud. PRP, right? So I wouldn't be here without PRP. I was studying bone marrow transplantation when PRP came on the scene. Whenever I hear PRP, I think inflammation. Can you tell me your experience with PRP and hair restoration?
Yeah, for sure. Well, first of all, my experience with PRP and hair restoration dates back over 13 years and 7,000 treatments. And in the beginning, we took our information from the wound healing folks, really using a simple tabletop spinner and test tubes, and literally pipetting PRP out of those test tubes into a syringe. Over the course of time, we learned that the methodology, if you will, that we would use, the protocol to separate the platelets, would make a huge difference in the results that we were seeing. And so after trying dozens and dozens of different types of kits and protocols, and being approached from dozens and dozens of different folks from all different suppliers around the world, I finally got frustrated and bought myself a Coulter counter to try to figure out exactly how many platelets were in the final PRP product, because I didn't basically believe any of the reps that were saying that they were the best of the best of the best. And I didn't believe any of the third-party information either. And so what we were able to do is essentially now come to a point where we've determined really what the sweet spot is in the platelet concentration. And I think I showed you that in the presentation. About 1.5 million platelets per microliter is our goal, and there's some good science behind that. And I think actually I learned some of that at Joe Krieger's conferences with some of the orthopods there many years ago. But the point is that there may be other components like monocytes and such, which add to the effects of the PRP. And monocytes have a natural anti-inflammatory component to it. I didn't really go into every single step of my PRP protocol, because obviously that's what our training programs are for. But we do everything from apply light therapy to topical growth factors, specific way of microneedling, and the depth of penetration, and how many cc's were actually, or fractions of a cc's we're injecting in each square centimeter of scalp, and so forth, to really get a powerful hair regrowth effect. And so when I meet my colleagues and they say, well, PRP has really been disappointing for me, I'm not really interested in PRP, I always ask them a bunch of pointed questions as to what kind of PRP they're actually making, because I've been through it all. And they usually have...
Because I'll get to how they're making the PRP in a second, but I want to know how you decide whether it's an inflammation, an autoimmune, or a genetic hair loss.
Oh, well, that's going to be from our diagnostic procedure, right? So if you're a male, in your 20s with a receding hairline and thinning in the crown, it's going to be primarily genetic. That's the DHT effect on the hair follicle, causing the miniaturization, right? The change in the antigen-telogen ratios, the length of antigens changing. So you're getting a thinner, shorter, weaker, wispier hair over time until the follicle finally is dead and gone, and essentially beyond repair. And so that's going to be very different than an inflammatory process where you may have a circular area, which autoimmune condition, or a problem like in women, we see female frontal fibrosing alopecia, where they're losing their hairline that's receding straight back, and you've got a skin tone change, or central centrifugal cicatricial alopecia, if you will, in African Americans due to the use of relaxing agents, and essentially a burn on the scalp over time, and a replace of the follicle with scar tissue. So sometimes you need to do a biopsy to differentiate whether you have a scarring alopecia or not. But the good news is that PRP often cools down some of those scarring alopecias and the inflammation for sure. The other thing, Duncan, is that we know that even in male pattern hair loss, there is some microinflammation that's going on. So there may be some great benefit to some of the anti-inflammatory properties of PRP in conjunction with those growth factors that are telling the follicle, hey, let's turn on and get it going. So you get this kind of dynamic effect where you have an anti-inflammatory and pro-hair growth effect. And I think that also happens with laser light therapy, to be honest, as well. You get an anti-inflammatory and pro-hair growth effect. So if someone's confused or concerned that they're not sure whether they have an inflammation or a hereditary pattern of hair loss, they need to find a physician to do that.
So PRP, mitochondrial DNA, which is inflammatory because it's bacterial, right? So I'm afraid that a patient comes in with Areata, and a doc will use PRP, and that will exacerbate the issue. That's what I think is happening. I think in the case of an androgenic, there are definitely growth factors there. They cause growth, and that is sort of logical. But that's how I've been looking at it. I'm not sure.
I've never seen one case of exacerbated inflammation from PRP in over 7,000 PRP treatments. I've never seen exacerbated inflammation.
I believe you, but I've heard it 100 times. But then maybe they're using leukocyte, maybe it's their process, right?
Yeah, and the other thing is that let's say, for example, their hematocrit is high. If they have a 2% hematocrit or more, then that's a very inflammatory PRP. Or if they're lysing the blood, red blood cells during the harvest phase, then you're going to get some significant inflammation there.
Yeah.
I think, Dr. House, what do you think?
Well, first line main treatment for alopecia areata, of course, is injection of corticosteroids. And we do know there's several articles in the literature that PRP can be helpful for PRP. So Duncan, that should allay some of your fears about that PRP is triggering or making it worse. We know from clinical studies already that PRP can be helpful for alopecia areata. So
yeah, I've had great results in my areata patients. I actually don't do steroids at all.
I recommend always doing steroids for patients because they can go to their dermatologist. I'm facial plastics, so it's not in my wheelhouse, but steroids are covered by their insurance, and let them go see their dermatologist and have it covered by the insurance, and patients are usually pretty appreciative.
Unless they didn't get a response or they have skin necrosis from it, then they're pretty appreciative that we have a good result.
Financially, it works well, and most dermatologists can handle a little low-dose steroid injections once a month to treat them with that, and it's highly responsive. So that would be a good place for them certainly to start for that, and then certainly if they want to continue with some PRP, that is definitely a nice place. And as we both know, it's been proven effective, and so that can work well. Also, I've definitely seen that PRP when it first came out, again, having started using it over a dozen years ago, wasn't so effective in the beginning. And definitely as the treatments or the preparations of the PRP have gotten a little bit more clarity as to better preparations of the platelets and the growth factors, the treatments have gotten a bit better. But definitely the combination therapy, combining it with our minoxidils, combining it with our laser light therapies, providing a better package, I think, to our patients rather than the standalone therapy. If I combine the two therapies against each other between PRP and exosomes, I think that if I had to recommend a single injection, to me, the exosomes are looking a bit more promising. I think what's really interesting, and I really am going to back Alan up in saying this, what really needs to be known are studies. We need studies. Please support us, doctors, in wanting to do these studies, because I think what would be really interesting is, what is the right answer? Is it to do exosomes once, to do exosomes every 6 months, to do exosomes in combination. I mean, these are the questions that patients want to know too. Should I do exosomes and then do, and Duncan, weigh in on your thoughts on this. Should we do exosomes and then do PRP every other month?
That's the question, protocol, because I actually have gone way past this recommending one thing. I think it's three things at once. I think it's platelet-rich fiber matrix, I think it's exosomes, and I think it's amniotic fluid at the same time, if you can afford it. And that's why we sort of give away our amniotic fluid now, because I want the outcome, right? But how many times are we going to do this? So the platelet-rich fiber matrix will keep it's going to cause a gel as soon as it goes into the tissue because there's calcium there for fibrosis, what you just did. And I like that approach. You're not going to convince me that PRP is not inflammatory. Whether you remove the leukocytes or not, it's definitely inflammatory. Now, maybe something else happens that causes an anti-inflammatory response, like ozone or extracorporeal photophoresis. But the use of all of these together and maybe waiting 4 to 6 weeks, and again, you have the experience, I don't. I'm just telling you that I have gotten here by just theoretically thinking this out, and it's worked. So I think that if you use those three products together, you get temporary anti-inflammation from the amniotic fluid. You get permanent growth and anti-inflammation from the exosomes, and you would get a holder from the PRFM. But then again, these two will negate the inflammation I'm talking about from the PRP, and then you get the growth factors. So it's complicated, but I do think that PRP is inflammatory. If you add the amniotic fluid, it's going to stop it.
Well, we know all PRP is not created equal, and we know that this is a similar paradigm to what they do in MSK, right? So you do dextrose, and then you do PRP, and then you do bone marrow aspirate. So you do these three procedures over, a 6-week period, and you do in conjunction with other therapies. So that's your longitudinal care plan pathway. So the one and done or the one size fits all. And if you look at other clinical areas like ED, which has similar inflammatory profiles from a patient standpoint, there's also an entire litany of mechanical and non-mechanical and biochemical and regenerative medicine and other injections that are used to help these patients, restore their sexual functionality. So I think what you're seeing is a combination of minds coming together, looking at problems, but looking at them in a very specific way. And that's really what the goal of this is to take people without clinical experience with biochemistry experience to combine them with people that have been in the trenches, learning what's best for their patients and actually dealing with patients on an individual basis. So as much as it may seem like it's disconnected, it's actually moving forward nicely, in my opinion.
Look at Dr. Hollis's pictures. Those look great to me. So, I mean, who's going to argue with that?
Well, Alan, how about you and I get together? We have different sites, and we take 50 patients, and we submit for. Let's go for an IND, and sounds like a good study to me.
Yeah, as long as somebody provides the product.
Sounds like a paper in the making.
We do give away % of what we make, so we're used to that.
But is that Duncan saying yes? Is that a yes?
If you give me data, I mean, I really have given away 50% of what I've made since the day I started, but I never get data back. So I keep trying.
Well, maybe you're working with the wrong doctors. I don't know. We provide a lot of data to our laser therapy folks and others. So, we just got to get on the same page in terms of what's, what we're trying to accomplish. And, sometimes we need to walk before we run. We've done smaller clinical trials and even, just for white papers and things like that before we went to larger, multi-center studies. So,
All right, maybe all right. So Alan and I are publicly saying that we will work together and come up with a protocol together. And
what works is you tell me the problem, and then I figure out how to fix it. But I don't know the problem. Like that's how the interaction.
All right, Duncan, you're gonna get Alan and me together, and we're gonna come up with a protocol. Get you big numbers for patients.
And you've done a great job. I started this with you 6 months ago. I'd love to see that data.
So we have a bunch of questions here, and Misti Barnes has just joined us. Thank you, Misti. We really enjoyed your presentation on trichology and scalp health, and try to get you involved in some of the upcoming talks. So, so we've got a bunch of questions. When do you typically see results for patients? Are there any benefits to adding light therapy after treatment? So maybe we'll just go quickly around, Dr. Bauman. You want to look at that?
I want to know that. Dr. Bauman, explain light therapy.
Laser light therapy, we now know exactly how that works. So the light energy at the red wavelengths is accepted at the photoacceptor molecule, the cytochrome C oxidase in the membrane of the mitochondria, and essentially it goes down the electron transport chain and you get increased ATP production. So that extra energy goes into making better quality hair, non-chemical, non-invasive, no side effects. And laser light therapy is actually your least costly treatment for a patient over time if they get a good device right off the bat. I've been using laser therapy since 1999.
How about you, Dr. Hallis? Do you have experience with laser light therapy?
Yeah, we had a couple of devices in our office for a couple of years, and often have our patients use at-home therapy, and they kept complementary to the things that we offer in the office, as long as you have a patient who's compliant with it.
Compliance is key, absolutely.
Yeah.
So
Without compliance, a lot of
what
this.
about the notion of patient expectations? What are we looking at before they see the peach fuzz? I mean, I think, is it within 2 weeks, a month, 30 days, 6 months? And I know it depends.
So we measure our patients in 90-day intervals, and that's a very, very specific amount of time because we know that hair grows at a rate of about a quarter to a half inch a month, or maybe about a centimeter or so. So when you have enough of time that passes and you've been compliant with the treatment, you can then use the cross-sectional trichometer to measure the results, and the cross-sectional trichometer, the hair check, right, over time. And it can predict how well things are going to look in 6 or 12 months. So at 90 days, most patients don't see too much of anything in the mirror. They don't look too different in general, but the measurements can sometimes change dramatically. As I showed in the presentation, there was a woman there who was in her 70s undergoing treatment, multi-therapy. I think she was using lasers and topicals. And at 90 days, she really didn't look all that much different, but her measurements were like off the charts, phenomenally good, like way above average. So I said, hang on, you're doing great. Keep on going. Your measurements are telling us that things are coming. And so at the 6-month timeline, the results were they looked amazing. And so that's what I showed in the presentation, 6 months. And then of course, 12 months later, it was a dramatic result. So if you're not measuring, you're not going to be helping the patient too well understand their baseline, and they're really not going to be understanding how their hair growth treatments are working over time. And also, if you've done a PRP or exosome treatment, you're not really going to know how long it's lasting unless you measure, especially if you've only taken a before photo. Sometimes you're going to get stuck where a patient comes back after they've lost all of the results of their treatment, and that's like no bueno, as we say here. We want to catch it at the plateau phase before it starts to dip down, and then readdress it and retreat it at that time.
Okay, our next question I'm going to address to Misti Barnes, because I think it's right in your wheelhouse. So scarring alopecia, can you describe some treatment options for that in hair restoration?
Yeah. Well, scarring alopecia is usually autoimmune based. Also, could be caused by fractional alopecia that advances into a more permanent scarring state. I treat people with scarring alopecia actually quite often. As I mentioned in my presentation, I think it's really important to look at what the causes are of things like fibrosis that's occurring within the scalp, calcification, and I have found microneedling to be one of the better tools, and that's why I combine it with peptides. So I find that if we're approaching the collagen matrix that's now dysfunctional, and we're treating it with using that wound growth trigger that we get from the microneedling, and we're advancing more collagen growth, that we can get some results. I think it depends on how long they've had it. If it gets worse, CCCA it's a long-term scarring, and what we don't want is that. But I still believe that even if they've got some deep scarring, the follicle base, that we can heal it. It's just a matter of how we approach it, what processes we use, and what type of peptides we're using. In my experience, what type of peptides.
Alan, you want to jump in on that a little?
So scarring alopecia, yeah, absolutely. For CCCA, central centrifugal cicatricial alopecia, it's a very yeah, easy for me to say. That can be a very dastardly condition, and it can wipe out a lot of hair. In fact, I just had a consultation this morning with a woman who had CCCA over the entire mid-scalp and crown of her scalp, basically nothing left except really occipital hair. And so it's very, very difficult to treat, and not always amenable to transplantation, because if the inflammation is still active, then it could kill off the transplants. So not too much fun. A lot of our cicatricial alopecias that I see in the practice are caused by plastic surgeons, actually. So we're doing a lot of scar coverage and camouflage in the sideburn area, areas that have been invaded through older facelifts and brow lifts, and even the newer endoscopic brow lifts can still leave patients with some scar tissue. Those patients are super happy with transplantation into those scarring areas. Absolutely, if you can find scarring alopecia patient that is an autoimmune or an inflammatory process, then PRP, laser light therapy, even some good nutraceuticals can start to calm that down. But you may need a more traditional approach as well, from antibiotics like doxycycline and such to hydroxychloroquine can actually help as well, in addition to topical steroid treatments. So if you have someone with scarring alopecia, I definitely recommend you send them to a specialist so we can take a look at it and see what's going on out there.
And Duncan, I think you began most of your experiences dealing with scarring as well.
I try to remind people is, I didn't come up with all these alternative uses of exosomes. The first thing I saw was, the abrogation of scarring in a burn victim. All of this was brought to me by these physicians here. All your idea, I just make an anti-inflammatory or immunomodulatory exosome. Very excited about the next exosomes. We have neural-specific, more anti-inflammatory, hair-specific that is actually going to be produced tomorrow. But who knows what's going to be better? Like, it has to be tested. I can't foresee. So the growth factors that are being delineated are important. The Maduro vaccine was sort of the best thing that ever happened to me, because now everybody has wrapped their mind around what an exosome really is. An exosome is the Maduro vaccine. It's a messenger RNA, encapsulated by a liposome. That's it. So if that's what it took to get people to understand what we're doing, then great.
Well, that's where our journey began, because I used you to teach everybody that it wasn't the cell that was differentiating into the tissue. There was actually a paracrine signaling effect. And it seemed like such a simple concept, but nobody really could grasp their mind around, I take bone marrow cells, I harvest them, and then I stick them into the inflamed area, and they become chondrocytes, or they become neurons, or they become, and people still believe that. It's on the FDA's website that these cells differentiate and become other tissue type. You were the first one who eloquently put out the notion of cell signaling and was able to prove it, not just with a product, but with a science. And now you're actually leading the way again by characterizing these with, at a great expense to you technically and, mentally, to put together the tools that the industry needs to know what these microvesicles actually are. It's a long way to go, because this is the beginning, right? So, Duncan, speak to this a little bit, because I'm trying to get through these questions. The difference between placental-derived exosomes and perhaps ones derived from like a young bone marrow donor.
I started this whole business of exosomes by growing people's cells, and that's the reason people didn't believe me when I started, because if you'd ever grown somebody's cells, you would never want to do it again. It is so difficult that when I realized I could get out of doing this, it was like, hooray. The older the patient's cells are, the more difficult it was for me to do it. And sometimes I would expend 8 months worth of effort and electricity and serum, etc., to be able to accomplish that. When I had a young patient, it was like this. So I started researching, why is that happening? And then I started finding papers that compared 40-year-old mesenchymal stem cells to placental mesenchymal stem cells. And as you can imagine, a baby's growing, so they express more growth factors, so the baby grows. It's really simple. And they laid it out by doing Westerns and mass spec on the different protein profiles of a 40-year-old versus a baby's exosome, and there were just more growth factors than the young one. It's not complicated. So that's when I went straight for placental. That's when all my outcomes started increasing. You mentioned amniotic fluid before and erectile dysfunction. I had a physician, a urologist, with 200 urologists under him, take me to dinner just to thank me for exosomes, because he had increased his outcomes from 80% with amniotic fluid to like 95%. Why? Because, first of all, they're specific, they're placental, and they're repeatable. And amniotic fluid is derived from epithelial cells, whereas ours are derived from sickle stem cells. So Here's a way to make it sound like I'm not just blowing smoke up. Wake Forest uses placental stem cells in their tissue engineering product. That's it. That's all I have to say.
Okay. What if we're getting a question regarding exosome treatment for beard growth? Who wants to handle that one?
Well, for beard growth, we've been using mostly hair transplantation, to be honest, over so many years. Anywhere that you have a hair follicle that's weak and thin and wispy, you can use these regenerative medicine techniques. In addition to other things like even minoxidil can work if the hair follicles are weak. But usually when we have weak beard growth, it's typically more of a kind of a patchy loss, meaning that there's just a lack of follicles in that area, from the get-go. And so, a transplantation is typically the best option.
Theoretically, if you microneedled me and injected exosomes, it should come out brown, black again. I have not experienced that, but I do have users that have experienced that. So maybe I'm just not causing enough inflammation. But the stem cell factor that needs to be in there for that improvement, et cetera, has been written in the literature, and I really want somebody to try this so you can explain it to me.
Kind of wrap up a little bit here, but we'll deal with the white elephant in the room. What are the regulatory complexities of using these for aesthetics? We know that aesthetics and regenerative medicine is somewhat not only the holy grail, but it's the low-hanging fruit. The more complicated we get after some of these indications, and we know people are looking at Parkinson's and autism, and God bless them, because they've really been out there trying to do what no one else will do to try to give patients hope, and that really is the essence of translational medicine, despite what the court of public opinion says. A lot of times these patients are actively looking, but we know that most companies are rushing towards the low-hanging fruit INDs and looking at these for cosmetic applications. So Duncan, do you want to discuss that a little bit?
Well, now that I have the experience, nobody's going to get approved unless they have a defined cell line. None of these amniotic or Wharton jelly, none of that is going to get approved. The FDA doesn't know what an exosome is. They do know what a cell line is. So that's all they want. They could care less about the 6 years of exosome-specific characterization that I have. They want your cell line characterized, and that costs about $150,000 to $200,000. So if you don't have a cell line because you have multiple donors because you're a tissue product, you can't achieve that. Ergo, you can't get your product approved. Now, topical products are always going to be okay because cosmetics are topical. So I see no issue with what we're talking about today in any way as regards to the FDA. I wish you hadn't even said the word autism because we are so far from that. And I'm actually expending a lot of effort in a neuro group. We're going to study Parkinson's, but we're going to do that the conventional way. We're going to do our secret phase 1, 2, and 3 and work through that way. It's not appropriate for that. But as far as we're concerned, we're talking about topical creams right now. That is really what we're talking about. And whether you microneedle it in there or push a needle it in there, it is still topical. It's not systemic. And that's the key word, systemic versus local. As long as it's local and you're an MD, it's within your purview, A. B, the FDA has a lot more to worry about. C, they actually don't work in the building anymore. They're all, working via Zoom. It's a different world right this second. That's one thing. The second thing is that the FDA extended regenerative medicine cutoff from November 2020 to May 2021. The last time I heard Peter Marks, MD, PhD, speak, he said we might extend it till 2023. What they're doing is they're stressing you out such that you get your IND in, but they're going to keep pushing it back 6 months at a time. So I would suggest that you relax until companies like myself or the others get something approved. But it's never going to affect what we're talking about today. Hair and skin has nothing to do with regulatory processes, as far as I'm concerned.
The bar needs to be at least established, as you had mentioned before, that the quality of the products, the laboratories that are producing them, the studies they're involved in, their commitment to the scientific process, and at least in the worst case scenario, don't infect anybody. Whether it works or not is irrelevant as long as you don't transfer E. coli.
You can't call your product an exosome if there's no exosomes in it and destroy the exosome market because you wanted to get people to buy your exosome. That's wrong. There are no exosomes in any of the products that I've characterized. But yet exosomes got a letter. There were no exosomes in that product. It was a contaminated Wharton's jelly product. But anyway.
And Joe, also to that comment that, with regards to the FDA, it's a lot about the claims that you're making. So, we have to kind of work around that. If you're making claims that are excessive or anything that they deem non-cosmetic, then that's when you're in some hot water. Hair, by the nature, for me personally, with peptides, I am careful about the claims that I make. I'm also aware that hair in and of itself is considered cosmetic. So unless we need a script or we're looking for a script and we're not making specific claims, generally the FDA is going to kind of ignore you.
And just as another commentary, I mean, as we discuss personalized precision medicine and longitudinal care plan, Alan, I know that you've come up with looking at the role of testing for your patients and actually supplementation as an adjuvant for therapies and care down the line.
Well, yeah, that's absolutely true. I mean, when we're looking at the scalp and there's some, poor hair growth there, we're taking a very holistic approach, as you saw in my presentation. Everything from risk factors to lifestyle to medications they may be on. If needed, we're going to do blood testing and really take measurements right at the level of the scalp just to see what the scalp health is doing, as well as how good the hair follicles are performing, and then tracking that over time is really the key component, because in this day and age of biohacking, just use that term kind of loosely right now, it's all about the metrics. It's all about the measurements. And so patients want to know how well they're performing in terms of their hair. And they know that it's going to be a long-term thing that they're going to eventually see in the mirror, but they want to know as quickly as possible how well their treatments are working and if they should make any changes. And, honestly, so do we. So it's a big part of it.
Okay, so I think that pretty much covers the bulk of what we set out to do, and I thank all of you for really contributing incredible content and intellectual, contributions to what makes a difference. And we have a great spectrum of intellects through all of you. We have the wily old veteran that's been out there plotting away for decades, Alan Bauman. We got the crazy mad scientist Duncan, who's been toiling away in the lab for different reasons and coming up with discoveries and now working hard to characterize and classify. We've got plastic surgeons working to help their patients' lives, and trichologists like Misti. All of you come together. You all bring a different element to the equation. But I think when you stand back from space and think about how is hair loss treated, how is it approached, this is probably the best it's ever been. And it really takes people like yourself, as well as industry and science. Patients have to be involved. Patients have to do their part to be involved. But I think that, the basket is full of tools and ideas to really help people, not only from a cosmetic standpoint, but people that have suffered from cancer and other disabilities that really make a difference in their lives. And I think at the end of the day, that's really why we all do this, right? Is to help our fellow man and to try to be giving of our time, talent, and treasure. So I appreciate you all coming here and giving me the opportunity to hang out with really smart people. I feel like my friends are really some of the smartest people in the world, and Boston Biolife is blessed to have you here, and we really appreciate all the contributions that you make, and we look forward to doing this again real soon. As mentioned before, Boston Biolife is providing complimentary webinars to our listeners. These are easy ways to stay current on new and emerging therapies. And typically, we fall into three categories. One is biotechnology, biochemistry of medicine. The second being clinical practice, which is the clinical applications of these technologies, and ultimately is practice management, how you run your business and how you maintain compliance and good practices for your patients to ensure the best possible outcomes. A deeper look at the MedPub research tool. As you know, these technologies are ever changing, and Boston Biolife is pleased to announce its relationship with EBSCO Publishing. This affords us tremendous access to the world's research literature database in real time on a wide variety of topics which transcend both biotechnology and clinical practice, so that you can stay current on the latest research and publications. These do come with CME credits and are a very useful way for you to stay current in your field and to learn the next evolution of your medical practice. Again, the MedPub Research Tool offers 20 CME credits, 9,500 peer-reviewed journal articles. It's an easy way to stay current on new and emerging medical technologies, best practices. It functions as a desk reference, and oftentimes you can use it as a patient educational tool in facilitating patient engagement. Industry access is an important part of what we offer at Boston Biolife. And with our relationships with our technology providers, we can offer you discounts on industry events, access to their virtual content, again, updates on research and development coming from the field, stay current on FDA issues, stay compliant with the resources that we offer, such as malpractice insurance and medical legal defense, as well as regulatory compliance evaluations. Stay aware of new and emerging technologies as they develop in the field, and to leverage some of the incentives of technology providers and their representatives and access to additional training. Some of the organizations that we're currently working with now, as we had in the past, and this will most likely evolve as well, so we encourage you to check back with bostonbiolife.com, is working with the AAOM, the American College of Regenerative Medicine, the Medicine Society, the Perinatal Regenerative Medicine Society, Total Orthopedic Biologic Works Institute, the World Academy of Pain Management Ultrasonography, and the World Stem Cell Summit are just some of the groups that we're working with that we can offer discounts for attendance and access to the resources that those organizations provide as well. For additional information, please send an email to josephkrieger@bostonbiolife.com. Feel free to call me at 978-569-8080 and visit us often at bostonbiolife.com.